Hello po. Pwede pong pa-rename sa iba. Sure.
Ay, NJ, mag-rename ka na. NJ.
Hello, good morning.
Hello.
Good morning.
So, of course, today would be our day one.
Mag-rename ka na.
Ano po yun? May naririnig na?
Naririnig ba ako ng maayos?
Yes po, Mami Jane.
Okay. Parang mahina mo.
Ay, medyo lang po. Opo, medyo mahina.
Check, 1, 2, check.
Ayun, better.
Okay.
Ayun. So, again, thank you sa inyong lahat. Today is supposedly a holiday, but we're all here to work for the love of our work. And so ang schedule natin is 8 to 9. We have the registration process. So we're really expecting them to enter the room muna. Pero okay lang, just quickly, ipasadahan natin paano tatakbo yung araw. Okay lang po ba?
Sure. Okay.
Yeah.
Yes po, sir. Share my screen. Nakikita na po ba yung screen? Yes po, sir. Yeah, thank you. Ayan, so ganito lang. For those na sinend ko sa group chat natin tong file na to. So kapag ka medyo nakalimutan natin kung nasan na tayo, just refer back to this document, no? Para level off tayo at alam natin kung nasan na tayo. So pasadahan lang natin, 8 to 9 will be our registration. Ang in charge dyan ay Secretariat, of course, and Tech Team, plus RAs at TAs ng Math. Sir Francis, MA, thank you. Thank you. Ayan. So of course, our job is to supervise everything. Yan. So kailangan on standby tayo ha, Secretariat at saka Tech Team. So maganda siguro kung merong sa bagay, pwedeng tayo na lang mag-usap dito, Secretariat and Tech Team, kung sino yung mga titingin sa FB. Sa FB, sa YouTube. Yan. We can just talk about the specifics later. No? Yan. So next, we have the opening program. Yan. Andito naman na si Sir Don sa campus. So I think he'll be joining us in a minute. Yan. He'll just give a short opening program that he will just welcome our participants. And then next, right after Sir Don, NJ, pakisalo.
Yes po, sir.
Yan. So for NJ, don't forget to, again, parang note, I think i-mention din naman ni Sir Don, pero emphasize lang natin, we would want to like apologize for our participants for holding this on a holiday. Yun lang kasi we're pressed for time. We don't have other days. So we really need to push through with day 1 today. Tapos for those who can't participate because they will be in the celebration of Eid al-Fitr, they can always refer back or they can always go back to the recordings naman sa YouTube. Yun lang, mga few reminders. And then etiquette and etiquette and general reminders, as I've mentioned kagabi sa email, you may use the file na ginamit mo nun kay Ma'am PJ kasi standard naman yun for every talk. Tapos, meron din lang general reminders na ginawa si Ethel. Ano naman yun, ifa-flash naman yun during break time. Tsaka kapag may ibang moments na walang magsasalita, ifa-flash yun. So anyways, you just work with the file that you have. Okay?
Yes po, sir.
Yan. Tapos introduce na lang ang ating star of the day. Yan, Ma'am EJ. Yan. Ma'am EJ po, after the brief introduction, you will have your lecture proper in 9:30 to 10:15. Yan. And then after nun, short break. Ayun. Ito lang pala, may I solicit for your opinions. Pagka short break, especially lunch break, pwede bang mag-leave group yung ating participants una?
I think they can. I think.
Pero discretion nila yun. Baka hindi sila makabalik. Tama ba?
Yeah, oo. Kasi baka mamaya pag kunyari, assume natin na mapupuno yung 300, better if nandun lang sila. If kaya naman nila. Pero kung hindi, syempre wala naman na tayo siguro control.
Hindi naman natin yung control.
Yeah.
Sige. So, okay lang kahit hindi na natin sabihin sa kanila yon, dapat they should understand that. Okay, next, may demo of cell culture ng 10:30 to 11:45 ulit si Ma'am EJ, and then open forum. Yan, NJ, ikaw ulit dito ha.
Yes,
kindly facilitate. Bale, kumbaga ikaw yung parang overseer for this event. So,
yes,
ikaw nang bahala mag-direct ng flow. Okay?
Yes, po.
Thank you. And then ikaw na rin magsabi na magla-lunch break ng 12:30 to 1:30. They may opt to, sige, sabihin mo na, i-warn mo na sa they may opt to leave the meeting, but please note that, hindi siya assurance na baka hindi makabalik.
So,
okay.
Sir?
Go lang. Yes,
Sir, sorry po. Sir, kung sakali po ba, pagbabanggitin na po yung break, pwede po bang banggitin na or parang i-request na lang po sa, specifically sa Zoom participants to not leave na lang po yung Zoom meeting?
Kung kaya nila.
Yes po. Sige po.
Parang if your circumstances permit,
Yes po.
to ensure na kasama pa rin sila dun sa Zoom. And then we will, thank you. And then we'll resume ng 1:30 to 2:30, mag-NJ ulit. And then open forum, NJ ulit. Okay? Short break. Ito pala. Sige. So after nung Q&A sa part na to, sa 2:30 to 2:45, crucial ulit yung role mo dito, NJ, no? Kindly remind them na may evaluation after the activity. Well, dapat sabihin dito ng facilitators, pero banggitin mo na rin dito. Crucial to kasi pagdating dito sa 2:30 to 2, after ng Q&A, some of them would be into breakout rooms. Mga nasa YouTube, wala na. Sir Francis, yung sa YouTube, pwede sigurong i-stop na natin yung stream.
Kuya Jano, sorry.
Yes?
Ano ko lang i-share ko lang na live na kasi tayo on YouTube. So technically, naririnig na to doon. So kung may nanonood na, naririnig na tayo.
Okay. Hello! Hello, participants. Welcome.
Oh, no. Anyway, yun lang.
Welcome. Nakita na nila yung... tama lang yun.
Pati yung screen nakikita na din nila. So...
Okay lang, wala namang sensitive dito. Pero sige. Okay. Anyways, yan, crucial lang doon sa 2:30 to 2:45. Kindly remind na lang for those in the YouTube, hindi na sila makapasok dun sa breakout rooms. So, they can still get the benefit of the activity by engaging gamit dun sa FB group. Pero dun sa mga nasa Zoom, sila yung pwedeng mag-enter ng breakout rooms. So, there they will be interacting with the facilitators. Yun lang, huwag lang nila kakalimutan, merong eval form. Ayun din po pala, after nung Q&A, yung mga nasa YouTube, pwedeng isend na dun yung link for the evaluation form. Okay. Tapos, balik tayo dun sa Zoom, sa breakout rooms. After the activity, facilitators should remind the participants na merong eval form. In line with that, saan kaya natin makikita yung yung links to the eval forms? Ethel and Steph, tama, meron na ba? May working link na? Okay, sige. So paalalay na lang na kapag kakailangan namin yung link for the eval forms. Okay, I think yun na yung summary ng day 1. Kung paano tatakbo ang ating day 1. Questions?
Kuya Jano, sino pong mag-i-introduce kay Sir Don?
Actually, secretariat sana. Okay lang, din.
Sige po. Ano na lang din, pag in-admit natin sila, magiging flight stewardess na lang din.
Oo.
Tapos reminder po, pa-change naman po ng names. If kaya. Para yun lang naman Kuya Jano. Thank you.
Okay. Sige, okay lang po. There's a request from our speaker. Check lang natin yung pag-share screen ni Ma'am EJ. Saglit. Okay. So,
Hello? Hello?
yes, Ma'am EJ.
Is it okay yung resolution niya? Is it
Yeah.
not?
Yes po, okay naman po siya.
Okay, sige, sige. Okay. Okay na. Stop share.
Hello everyone, good morning. And good morning everyone. Thank you very much for sparing your holiday time this Thursday. So kung may mga kasamahan po tayong mga Muslims, happy Eid al-Fitr po sa inyong lahat. Anyway, thank you very much for participating to the 26th Summer Institute in the Natural Sciences and Mathematics under the biology discipline. So thank you very much once again. So while we wait for the other participants to log in via Zoom, may we request our participants to at least mute their microphones for now so that there's no feedback and all. May we also ask our participants if they can rename themselves, but if you can't, we will be the ones going to rename your Zoom meeting name. So we have 5 minutes left before we formally start our program for this morning. So as you can see sa ating mga participants po for today, I have been emailing together with my SINSM Secretariat team, Mr. Brian Martos, the details of our event for today. And I have been we have been inviting you to join our Facebook group. So for those people who are not yet members of the official Facebook group for the 26th SINSM under the biology discipline, we are encouraging everyone to please visit your email. So sometimes napupunta po siya sa spam section ng inyong email. So kindly check if nandun po yung inyong mga, nandun yung email nyo from the SINSM Secretariat. And also you might want to join the Facebook group because we will be posting updates and announcements. And you can also interact with our amazing lineup of speakers under the biology discipline for the 3-day event. So welcome everyone. I hope nakakain po tayo and ready po tayo makinig sa mga sa ating mga lectures for this morning and later this afternoon. Good morning. Ayan. So for our participants, kindly put in the chat box naman kung nasaan po kayo right now. Currently, of course, this is an event of the University of the Philippines, Baguio. So nandito po kami ngayon with the whole SINSM team of the biology department. Nandito po kami lahat ngayon sa Baguio. So yung mga participants po namin, kindly drop your location on the chat box or on the comment section because we're live via YouTube. Ayan po. So if you have questions as well, and if you're watching via the YouTube live stream, kindly drop your location in the YouTube live stream. Ayan. So ayan, we have participants from Isabela, we have participants from Cagayan, Dagupan City. Sana hindi po kayo masyadong nasistress dahil sa init dyan. Napakataas po ng heat index natin dyan ngayon. Ayan. We have participants from Nueva Vizcaya, from Laguna, from NCR. Hello po. We have also participants from Pampanga, Tarlac. Oh, my home province, Bataan. Hello po sa mga taga Bataan natin. From Cagayan, from Negros Occidental. Hello po, good morning. Ayan, we have also participants from Tagaytay, Nueva Vizcaya, Pampanga, Las Piñas, Quirino. Ayan, sa mga kapwa ko Ilocano, welcome po. Sa mga taga-Vigan, Ilocos Sur, good morning po. Ayan. Hello po, everyone. It's so nice to see everyone virtually. Usually we conduct the SINSM face-to-face. Of course, we invite participants from all over the Philippines. We invite them here at the UP Baguio campus to, of course, learn from our amazing speakers and also, experience the Baguio City culture and also experience the UP Baguio culture. But right now, because of the current circumstances that we are in, medyo hindi po possible na mag-face-to-face tayo. So we still did our best to conduct the SINSM even via a virtual meeting. So, hello po. Oh, we have participants from Agusan del Norte, Mindanao. Hello po. So for those people who are having difficulties for their Zoom virtual background, you can actually reach out to our tech people via the chat function. Pwede niyo pong hanapin yung mga kasamahan ko sa SINSM team na nakalagay SINSM Tech or SINSM Secretariat. They can assist you with changing the virtual backgrounds that we have provided. Kung nakikita niyo naman po ang ating magandang virtual background for today. So mamaya, we'll be inviting everyone for a virtual photo op. So if you can change your background to what we have provided last night, and kindly change your virtual background po. Also, there are people asking if they're required to turn on their cameras. It's okay if you can't, especially if there are internet or bandwidth connections, it's okay. But if you're going to speak later or ask questions to our speakers, it's maybe proper to switch on your cameras para naman din po makita namin kayo. Hello po. So we'll start in a few. We're just waiting for the other participants because we had a huge number of participants this year and we're quite overwhelmed with the number and with the support and your interest for this event. I hope that you'll be able to learn a lot from our amazing speakers for this 3-day event. Again, if you have concerns, feel free to reach out via our Facebook group, or if you have difficulties with your tech, kindly message our SINSM Secretariat team or the SINSM Tech team. You'll see them within the Zoom meeting. So, and there are still people coming in. I hope you have. I think we have participants also via the YouTube stream. So let me just check. Ayan, meron din po tayo mga participants via YouTube live stream. Yeah, we have participants from Ifugao. Hello po, good morning. We have participants from Davao. Hello po, good morning po everyone. I hope everyone is safe. So we will formally start in 2 minutes. So I hope everyone's excited to learn. The session is recorded. So later on, we will be providing recorded clips of the meeting so you can access it anytime. Maybe you would want to use some of the resources here for your classes or maybe for your professional development. We will be providing the resources for this event later on. So make sure that you have access on our Facebook group. Again, check your emails, check your inbox or your spam folder for the email from our team. There are instructions then for you to join our official Facebook group. So, hello po, magandang araw po sa lahat. Also, just an announcement for those people who are into social media. If you have social media with you, if you have Facebook, Instagram, or Twitter, kindly put tag us, SINSM, or maybe put some hashtags. I think we will be showing a list of hashtags later that you can use for us to be tagged via Facebook. So kindly tag us on your social media posts para din naman makita namin yung inyong there you go. So tag us in your stories or in your posts with the following hashtags. Hashtag SINSM, SINSM 2021, Biology SINSM, and hashtag UPBAG. So tag us via Facebook, Instagram, and Twitter. So, hello po. Magandang araw. Yan, nakakakita po ako ng mga participants na gumagamit ng ating virtual background. So if you can switch on your videos, if wala naman po tayong limitations pagdating sa ating internet connection, kindly switch on your camera para din po may nakikita ang ating mga speakers later. Also, it's nice na merong interaction. So it's nice if you can switch on your camera so that we have at least, interaction with one another. Hello, good morning po. We will start in a few. So maraming mga high school teachers. Hello everyone again. For those po na merong difficulties pagdating sa pag-change ng inyong virtual background, kindly approach our tech team, the SINSM secretary or the SINSM tech team for your concerns. Pwede po nila kayong tulungan regarding the virtual background. Yeah, there. You have to click the arrow next to your video. Then there's an option there to click or to choose your virtual background. So, and then try to click po yung virtual background na in-upload po namin last night. For those using po their mobile phones or cellular phones, it's okay po if you don't, switch on your cameras. Maybe you're conserving your battery life. It's okay. But then for those people who are using their laptops or their tablets, if you can switch on your videos, go ahead po. So we have, interaction and you can see your amazing faces. Again, good morning, everyone. We'll start by 9:25. We're just waiting for the other participants so we can formally start our program. Ayan yung mga kakapasok lang po. Kindly drop on the chat box kung nasan po kayo right now while participating on the 26th SINSM. So, nasan po kayo ngayon? Kasalukuyan po ako nasa Baguio City ngayon. So, medyo malamig pa po pero maya-maya iinit na. Nasan po yung iba nating participants ngayon? Kindly drop your location via the chat box. For those people on the YouTube live stream, kindly drop your location as well. Oh, we have people from Cebu, Cavite, Quezon. It's so nice to meet people from all over the Philippines. With that, let's formally start our event for this morning. So welcome, good morning, everyone. Welcome to the 26th Summer Institute in the Natural Sciences and Mathematics with the theme Upgrading Senior High School Science and Mathematics Education, Content and Competency, Part 1. So for the biology discipline, we have the sub-theme Genetics and Cell Biology at the Crossroads of Different Biological Disciplines, with a general objective of reinforcing pedagogy and research in science mathematics through innovative approaches. Welcome everyone to the 26th Summer Institute in the Natural Sciences and Mathematics. So again, good morning po sa lahat. It's been a while since we conducted this event because last year was the height of the COVID-19 pandemic. So we weren't able to conduct the SINSM last year, but right now we were able to conduct the event for this month-long activity, and this is via virtual means. So again, good morning to all participants. Good morning to our speakers. So let me now introduce our guest, our speaker for To welcome us everyone in this activity, we have here our department chair for the Department of Biology, College of Science, University of the Philippines, Baguio. Please welcome Professor Roland Hipol, Chair of the Department of Biology. Please give him a virtual round of applause.
Maraming salamat, Sir Dane Soriano. Thank you very much for initially welcoming all of these participants that we have. I'm very happy and very honored actually to be able to welcome you formally into this occasion, this historic occasion. This is the first time that the SINSM will be conducting its activity virtually. Of course, this is not by choice, but it is by necessity. However, yung sinabi na, yung isang yung mga sinasabi ng mga scientists na when necessity is one of the drivers of innovation, and this is one, I think, one of the great opportunities for us to innovate in a sense, to make use of the available technologies in order for the SINSM to happen. And on behalf of the entirety of the Department of Biology, we would like to welcome all of you, all of us, 30 strong faculty, lecturers, RAs, and full-time faculty of the department would like to welcome everyone to the 26th Summer Institute in the Natural Sciences and Mathematics under the biology discipline or biology department with the theme genetics, cell biology, and cell biology at the crossroads of the different biological sciences. The Department of Biology would like to wish and hope that all of us are doing well despite the limitations or the difficulty, the tragedies that may have befallen us. However, we have proven time and again that we are all resilient to many of life's challenges. And this cynicism right here is a proof that despite the difficulties and the limitations that we've had experienced, many of us are teachers, and the academic years is continuing. In some schools, maybe it has already finished, but we have experienced the difficulties of trying to continue with the education of our students, and we truly care about the learning and the things that our students learn, such that we ourselves have innovated our mode of teaching. Of course, our line agencies, our schools, our administrators have provided for us maybe the necessary help or the structure with which we can use effectively to continue on teaching. However, the onus is with us as teachers to actually implement these modalities that are available to us and to imbibe and flavor all of these available modalities with our own inputs. And that was the challenge that we had. Di ba? And congratulations to all of us, to all of you who were able to uh, take care of business, kumbaga. And more than that, you were able to, well, hopefully our students were able to learn many of the things, hopefully all of the things that we planned for them to learn for this school year. Okay, so this is the humble contribution of the Department of Biology to these efforts, to your efforts as educators, as academicians, as teachers, as mentors. We would like to contribute to that treasure of information, of tools, of skills that later on you will be able to impart onto your students. The cynicism being the 26th at the moment, the 26th iteration of this activity is replete with historical stories. I hope many of the participants, the current participants at the moment, are repeat participants of cynicism, and they can witness to the many things, hopefully many things, that you were able to take home from the previous cinesums that we conducted. Of course, unfortunately, we did not have the 2020 cinesum because of we were not ready then. So we were ready actually. We were able to implement it, but we decided not to push through with the 2020 cinesum because we we recognized the difficulty of converting the face-to-face preparations that we've had into virtual presentations, and similarly we also recognize the difficulty of you teachers, academicians, mentors, and you might have had the difficulty of attending. So parang makikita nyo na missed opportunity yon kung tinuloy sana namin yun last year. But anyway, that's beside the point. So today, this time, we are ready, you are ready, and we are all excited to conduct again and to provide you again with the service, the help that you may need. Especially in this particular case, we will be looking at genetics and cell biology at the crossroads of the different biological sciences. And the theme for the Department of Biology is in similar ways that is the way by which we determine the themes of our CNESMs for the biology is that we look at the evaluation tools that we receive at the end of each iteration of the CNESM. And we seriously consider the suggestions that you forward to us. So in the 2019 CNESM, we had the botany cluster, or we had the botany cynicism. So in the previous also cynicisms, we had requests for genetics and cell biology, and that is the reason why we decided as a department to offer and to provide um, training or activities or for resources that may be of use to you as you will be discussing uh, the topics on genetics and cell biology in your classes. And well, one unfortunate thing about this being virtual is the activities will also be virtual. So unlike when the activities would have been face to face, so meron tayong mga microscopes, so we may have uh, molecular or equipment that can and will aid us in your understanding, our entire understanding of the concepts of genetics and cell biology. In the same spirit, we look forward to looking at your recommendations as you have finished after 3 days, as you have finished all the talks, listened to all the talks, participated into many of the activities that are prepared for you by our facilitators, our lecturers. Please provide us again with feedback, because this is definitely a source of inspiration for us, in a sense that we will be planning for the 27th CENISM. So as early as now, maybe as early as Saturday, once we receive the evaluation tools that you would have had forwarded, we will take a look at this seriously and determine the best ways forward. So kung gusto nyo ng, kasi in general terms, nakita naman natin dun sa CENISM, mismong CENISM theme. This is reinforcing pedagogy and research in science and math approaches part 1. So baka naman, kayo, kung ano yung ma-feel nyo na kung gusto nyo may part 2 ng cell biology or something else. So we look forward to that. Okay. So we definitely look forward to having you for the entire 3 days. And we would appreciate a lot if you would be actively participating in many of the activities that will be presented, that will be facilitated by our active junior faculty. So again, in behalf of our dean Professor Dimna Javier, the Dean of the College of Science, and on behalf of our Chancellor, Chancellor Corazon Abansi, and I, Roland Hipol, on behalf of the Department of Biology, would like to thank your presence and your continued support. Despite all of the difficulties, again, as mentioned, we are all here, and may we share a productive and learningful 3 days with all of you. Maraming salamat, and we hope you will enjoy and we all keep safe, every one of us. Thank you very much.
Thank you very much, Sir Don, for that very insightful and very inspiring message and welcome remarks. Ang dami nga po nating pinagdaanan since last year. And I think one of the commonalities that we have as educators is that we're really inspired and we're really driven to, give positive change to our students and to other individuals in our community. So thank you for that, Sir Don. Sana po makasama po namin kayo in the future events like SINSM po. So thank you very much, sir. So with that, hello again, everyone. Good morning po sa mga kakapasok sa ating Zoom meeting in our YouTube live stream. So just quickly before we proceed with the next part of the program, can we just switch on our videos for a very short photo op? Ngayong medyo mabilis pa yung ating internet connection. So if it's okay with everyone, can we just switch on our videos para naman kasama natin ang department chair ng Department of Biology. So we request our SINSM secretariat and team and all of our participants to switch on our videos. Can we just stop the screen sharing for a while so that we can get a photo. Ayan, ang gaganda nga po talaga natin sa ating mga feed ngayon. Napakaganda ng ating virtual background na ginawa ng ating SINSM team. Magandang araw po. So let me just take a few screenshots of our Zoom meeting right now. So, ngiti po tayo. Okay, first frame, smile po. Next frame po tayo. Ayan, napakaganda nga naman po ng ating umaga ngayon at napakadami nating matututunan for this day. Okay po, smile po tayo. Next frame po tayo. Smile. And our last frame. Okay po. Thank you very much, everyone. Okay na po kung kaya pong i-maintain yung naka-switch on na camera, it's okay. Pero for those with bandwidth connections, okay lang po na naka-switch off po tayo. So with that, let me introduce you to the next part of this program. So we have here my colleague in the Department of Biology, and of course, part of the SINSM team for this year, we have here Sir Noel Galido to introduce our net rules, our netiquette rules, and other reminders, and also to introduce our main speaker for this event. Take it away, Noel.
So thank you, Sir Dane, for that introduction. So hello once again to everyone. I am Noel, and I will be your moderator po for today's series of lectures. So before we continue po pala, we would like to extend once again our deepest apologies for conducting this workshop or this training during a holiday. Now to our Muslim brothers and sisters who may not be able to attend today's workshop, do not worry, as we may be able to provide to you or to those who may not be able to attend a recording of this Zoom meeting. Okay, now before I introduce to you our guest speaker, let me just go through a few preliminaries or some final reminders po. Okay, some final reminders which may be applicable to the succeeding workshop or trainings. Okay, so first po, the entire proceedings of this webinar will be recorded for documentation purposes. Also, all attendees of the webinar, except for the moderator and speakers, should keep their cameras switched off and their microphones on mute during the webinar and the subsequent open forum. Now, in case the webinar is interrupted due to a technical problem, all are asked to wait for 10 minutes to give the meeting hosts time to resolve the problem, or in case the problem cannot be fixed, to announce that the webinar has been suspended and will be rescheduled. Also, during the open forum, all those wanting to ask questions should send their queries via the chat feature of the Zoom meeting or in the comment section of the YouTube live streaming. Now, the technical team will determine which among the questions sent will be read by the moderator, yours truly, and the order they will be asked. The moderator will also acknowledge the member of the audience who fielded that particular question. And as such, participants are therefore requested to introduce themselves and their affiliations. The moderator will also determine if follow-up questions can still be accommodated or not. Now lastly, after the webinar po, participants are enjoined to accomplish an evaluation form. So later on, we will remind again everyone to accomplish the evaluation form, which will be posted this afternoon. Now, the link of the said form will be posted on the chat feature of the Zoom meeting and the comment section of the YouTube live streaming. Okay, so please let us all keep in mind all of those final reminders. Now, as we have all been prompted, our speaker for today would be Professor Saho. So Professor Maria Easter Joy Saho is currently a newly appointed assistant professor at the Department of Biology, UP Baguio, where she also finished her undergraduate studies. She finished her doctoral degree in medical science at Yonsei University, Wonju College of Medicine, South Korea last February, the year 2016. Now, during and after her graduate studies and postdoc stint at Yonsei University, she trained undergraduate and graduate students, interns, faculty, and researchers on primary cell cultures and biomedical techniques. Through the years, she has acquired technical skills on cell line propagation and maintenance, primary cell dissociation, isolation and propagation of 2D and 3D mammosphere culture, cell line transfection, and cellular and molecular biology assays, such as toxicity assays, immunological assays, and apoptosis assays, to name a few. She has also published numerous peer-reviewed journals and has presented her work on various national and international scientific conferences and meetings. To date, she is the treasurer of the Philippine Society of Cell Biology, or PSCB, in the Philippines, and a member of the Australasian Biospecimen Network Association, or ABNA, in Australia. Her exposure to these outstanding scientific communities heightened her passion for research, particularly on medical science research and those that can contribute to the development of her own country, the Philippines. So, to our participants, let us all welcome our speaker for today's series of lectures, Professor Maria Easter Joy Sao.
Okay, so thank you for that wonderful introduction. So may I know if my screen is okay? So can you see my screen?
Yes, ma'am.
Okay.
Yes, ma'am.
So again, a pleasant morning to all of you. Good morning, everyone. So hope you had a wonderful holiday today. So thank you for that wonderful introduction, Noel. And again, I'm Maria Easter Joy Saho. I'll be your lecturer for the whole day. So I hope I will not bore you that much. So as mentioned earlier, I am currently appointed, so just last semester, as an assistant professor here in UP Baguio, so under the microbiology cluster. So once again, I welcome you all to SINSM workshop natin. And this year, as mentioned earlier, our theme is strengthening the senior high school science and mathematics education, content and competency. So I hope we will achieve our goal this week. So our DB team or our department team will be the cellular and molecular actually biology and genetics at the crossroads of different biological disciplines. And the objective, as I said earlier, so it's reinforcing the pedagogy in teaching and also in research to science and technology or mathematics through innovative approaches that can be applied here in the Philippines. So today I'll be focusing more on the cell biology part. So here, and then tomorrow, there is another speaker. So Sir Madison will be focusing on the molecular biology part naman. And then on Saturday, Sir Geno will be focusing on the genetics part. So just let me just walk you through for my today lecture series. So first, I will have this lecture. So it's more of like an introduction and just a know-how on what is the applications of cell biology in different disciplines, not only in biology, but also other disciplines. So next, after this, we'll have a demo. So I'll be introducing a platform for you maybe that you can use in teaching. So we'll be using a virtual platform that can help you teach the kids today, especially, of course, it's a different era nowadays. So maybe we need to also innovate of some sorts. And lastly, in the afternoon, we'll be having another lecture on different techniques and models that can be used in research, not only in cell biology, but maybe like in other aspects as well. So I wanted to focus on cell biology because, of course, it's my expertise. And second, of course, who would not fall in love with this lovely picture here? So this is one of the picture that I took. So it's a A549 cells, which is a lung cancer cells that has been treated with CSE or cigarette smoke extract. So today, let's move on, start. Okay, so let's start this morning with an affirmation. So cell and molecular biology at work, and that's you, or actually that's us. So we are both wonderful and amazing. We are a wonderful creation, and then because you are just unique in so many ways. So we are amazing because we are made out of 37 trillion cells. So imagine that, and each of that cells have different in types or in characteristics. So there are 200 different types of cells, and then we are composed of 50 billion fat cells. So imagine that, so many fat cells, right? So 2 billion heart cells and a whooping 240 billion liver cells. So most of the cells have features common in our cells, like in the animals and also the plants in the world over a great white whale to a tiny moss plant. So every single living thing. So we next, like for example, when we go out, watch a wildlife film and spend few seconds visualizing all the plants and animals as cells. So we can see all of them, it's trillion and trillion of cells. So next, so we are also amazed because within our cells are working every nanoseconds of our life. So every nanoseconds our cells work, okay, not only us. So even if we are sleeping, so our cells still work. So from when you conceived and then until now, okay. So now welcome to the world of cell biology, where it's a city where all the fibers along with every minute molecules in our organs or in our system, there are different like motor signalings or like transporters. So it's like the jeep buses that are happening also inside the cell, like moving around, working as well. So here we have different parts of the city as well. So just like this inside the cell, there are energy converters. So what we call the mitochondria, right? And then they turn energy into the food and then chemical and then the electrical energy required by the system or by our cells as well. So for example, in our muscle cells, the energy is required to make it contract and operate as well. So I know you all know that. So it's just an opening or opener for all of you so that we will not get bored. Okay. So next, here is a figure of animal cells. So these are stained just like the one I showed earlier, but this is not my picture. So it's stained with a fluorescent label that help you visualize the cytoskeleton here. So the green one is the microtubules, and then the red one is the actin filament. So somehow it shows the structure of the cells. And then here, maybe stained in DAPI. So it's the nucleus where it's stained blue. So The cytoskeleton is usually not shown in simple diagrams of the cell because it's very complex meshwork or mesh like a strand. So these cells could not be cells without their cytoskeleton. So the structure will not be there. And then also here, the second figure, you can see here, it's a filamentous cells of a bacteria or bacterium, Rhodobacter spheroides. So the tubulin here, like tagged with CFP, that forms like a dot like structures, and then it forms somehow like it spirals in two distinct populations. So you have the green, the like magenta, like here at this side, and then we have, I think, cyan color, and then these are like cell bodies that are inside the bacterium. So here you can see just the inside of a bacteria and also animal cell. And then next we also have, of course, the plant. So these are Arabidopsis in root hairs of the plant. So the study of root hair in biology, especially in Arabidopsis, has provided a model cell type for insights in many aspects of plant development and cell biology. And also, it has been thoroughly studied to understand how plants regulate cell fate, like what will happen to this type of cell, and then the growth or in response to different environmental signals. So it's not only for animals, it's not only for humans, but also plants. So remember that all living things are also composed of cells. So cell biology in an exploration and the study of life at the level of the cellular level. So the parts that make up the cell and how these parts function at the molecular level. So that's what we usually do, right? I suppose you are all, or most of you are teaching biology or maybe cell biology in college if there are. So it's also the study of how the cells interact with one another. So as you can see here, it's not alone. Actually, when you see it in the culture inside a flask, so if one cell, one cell will not survive there alone. So usually it forms connection. It networks to another not another cells as well. So just like us here, so we're now connecting, networking, and learning with each other. So here, it is also the study of how cells interact with each other and how the normal growth of cell is controlled, and like how are they dividing, how they pass the genetic information after dividing, and then how they die as well. Or sometimes they do have that process of cell death or automatic or like autophagy or even apoptosis. So it is also part of a lot of field or study now that has been emerging. So the study of how cells move as well, and then obtain and release the use of energy and communicate and the signal to one another. And also cell biology is the center of life. And the critical center of life is, of course, you yourself. So welcome to cell biology. So here's just something that you can think about while having, well, hindi pa tayo start. Well, we start. So maybe while we have starting, so you can think about if you are doing some research work on cells, for example, some mouse, to help understand the cell biology of some maybe endangered species such as whale. So for example, you wanted to know maybe the fertility or maybe some fertility issues maybe now of the whales. So how could you explain why are we using, for example, these mouse cells? How can we use these mouse cells to someone who thinks only of whale as an enormous like animal or living thing in the sea? And then a mouse, a tiny. So how can we connect that? So it's just a tiny mammal that can be a pest. That's actually a lot here in the Philippines, right? So maybe you can think about that. How can we connect this, for example, like a specific mouse model into like a specific topic like endangered species of whale animals? Okay, so let's formally start. So today, just the outline of my presentation. So we'll be starting with background, just a brief history, and then the requirements that are needed by the cell, like the cell environment, physical characteristics that are needed. So the standard cell techniques, but this one will be doing it as a demo later on. And then some issues on contamination. And then we'll be discussing a little bit of different types of cell culture, because we have immortalized cells, but we also have primary cell culture that are also from um, directly from like either animals or even human. And then we'll introduce an emergent topic nowadays, which is 3D cell cultures, like spheroids, organoids. Like there's a new study actually nowadays that like there's this organoid that they have made, and then it made, or that organoid can have the ability to cry on itself. Okay, so that's a little bit just side like new study. So cell culture also as like in different industries. So we have also like in the commercial setting. Also, we will discuss some problems and limitation with this cell culture, and of course the application. So the wide application in its core life sciences and also like other applied sciences as well. Okay, so let's begin by defining what cell culture is. Of course, cell culture is a term that refers to the culture of disaggregated cells, while the organ culture describes the use of a non-dispersed tissue. Both encompass the description of tissue culture. So now, like here, cell culture, like some of the basic terms that you will hear, of course, cell culture, then tissue culture, cell line. So cell line are usually the one that you passage, or like the one that you will cultivate to propagate a little bit more, okay? So continuously. And then we have a finite cell line and then a continuous cell line. So finite cell line usually are the primary cells. So they have a timeline. So like until like this generation only, or what we call it passage. And then continuous are the immortalized cell lines. So that's something that they had tweaked, or they do some like h-tert or like tweaking so that they will live longer. And then next is primary culture, of course, are the one that are directly from like an organism, and then they culture it. So usually it has a timeline, as I said earlier. So like just a few passages. And then, of course, clone is the progeny isolated from a single cell. And then, so you clone the same cell. So immortalization, I mentioned it earlier as well. So lag phase of the growth is the initial phase. So nag-a-adjust pa lang siya. And then log phase is the growth that where it's in its peak. Okay, so most rapid growth phase. And then, of course, the plateau, or when the cell becomes confluent already, and then somehow it stops growing more and more. So population doubling is a time when the cell numbers, of course, are double. So cell banks are the repositories of the cell lines. And then we have substrate, passage, and then confluent. So later on, we'll be somehow like hearing all those terms again. Okay, next. So just for the history and background, so as early as 1895, is it clear? Maybe, yeah, if you cannot see, I'll just mention it. So as early as 1895, the first established method on cell culture was done using this, was developed using chicken embryo. So it was maintained for in vitro for several days. And then in 1989 here, they used the man skin cells. So they first used skin cells and it maintained in vitro. So the media they used here are ascitic fluid or acetic fluid, like from the, I think, ovary. So they use it as like a conditioned media. So next, and then eventually they develop a longer time to maintain it, and then they developed another functional experiment here. So they first had functional experiment, something that they can use in the future. So like the first technique in using lymph clot, so in 1907, so from a frog nerve, so here, so they get a frog nerve and then they grow it, and then they they develop a technique that can that used like nymph clot to somehow check the nerve of the frogs. And then next is yung development ng here si Lewis in 1911. So they first investigate different factors like growth factors in the medium that are required for growth, so or survival as well. And then next si Ebeling in 1922. So they first culture epithelial cells. So first they use like skin cells and other like animal nerves or tissues. But in 1922, Ebeling first developed the epithelial cells. So actually, epithelial cells, as I experienced, it's very hard to somehow culture. But they had developed the first culture in 1922. So next is they developed the cell line already in 1943. So about a lot of maybe how many years? 20 years, less than 20 years. So developing a cell line which is somehow immortalized that can last forever or a long time is a little bit, of course, during that time it's a bit challenging, but they have developed a way of maybe changing some of the genetic factors inside the cells. And also the next one developed Gay and also Earl developed the rodent cell line, and then Gay again developed the first continuous cell line, which is HeLa cells. So if you have maybe watched that drama on the immortalization of HeLa cells, so I think it's a movie or drama, and there's also a book on that they use this Black woman cells, and then actually there's some ethical issues there, but eventually they have I think they just develop the cells without them really like as consenting the patient. So anyway, after some years in 1965, so they develop a lot of use of cell lines. They developed a lot of functional assays as well. Okay, so here we'll just be quick in the next few slides. So here are the major cell banks that are available. We have ATCC. I'm not sure if you have heard it, but this is mostly where cells come from, especially if it's from the US. And then here also in the Philippines, we also get ATCC cells, but it's a little bit challenging to procure them. So at first, maybe 2 to 3 years ago, we don't have a distributor or local distributor for this. And these are very, very critical in terms of the shipment and transportation, of course. And if it gets stuck in the customs, so it will just die. And sometimes they don't provide like warranty, but good thing like ADCC, even though we have a lot of complaints already for the customs, but they always give us a warranty every time we purchase. Okay, and then next is this different cell types. So as I mentioned earlier, so we have the immortalized cell lines. So these are comprised of a single type that can be serially propagated, or that either for a limited number or cell division, so approximately 30 or sometimes indefinitely. So sometimes you have like, so we have this like first generation, second generation. So when you split the cells, so after the first generation, we call it like the second generation would be the passage 2. So the second passage and then the third passage and fourth passage. So you can split it into like until the 30th generation, but usually it's not advisable to use higher passage cell lines because somehow they have different characteristics already. So they have gone through a lot of stress and all that. And then next is primary cells, of course, is derived directly from the excised tissue, either of an animal or maybe a human sample, human tissue. and then following the dissociation into a single suspension, and then enzyme digestion. So next is another interesting type of cell, which are stem cells. So it's also an emerging field nowadays. So it has been a popular cell model used in research due to their unlimited growth characteristic and plasticity. So these cells are very unique. They have ability to self-renew themselves or to differentiate to another cell type or another type of cell in response to like appropriate signals within the body. So mostly some regenerative studies or even developmental studies, or here like tissue repair, maybe wound healing or replacement or something like that. And then next is 3D culture. So the recently the most recently emerging field. So it has a potential to bridge the gap between the in vitro and in vivo experiments. So again, in vitro is the experiments that are outside the human, or something like outside, not the physical. So it has a different and a controlled environment, while in vivo is like inside like a model. It's either an animal model, or maybe an animal, or it could be like a zebrafish or mouse, or maybe insect model. So while obtaining the results that reflect this in vivo condition. So somehow this 3D cell culture, it mimics closely the somehow clinician point of view, and also in vivo experiments or clinical, experiments. So physical environment. So these are some of the tips, and also the tips and tricks, and also things to consider or requirements in cell culture. So unlike just like other microorganism, like also like in microbio, so you need to have an ample or a specific pH that can cater the environment. So the pH of these biological liquids should be in neutral pH to permit survival of the whole animal. So for cell survival, usually it's from 6.6 to 7.8. And then for the optimal growth, it's usually 7.2 to 7.4. So neutral conditions. So that's why we have to monitor the pH of the cell. It's very important to know the pH of the cells for their growth and survival. And then also temperature. So usually we have this one, 36 to 37 degrees. So just like the temperature of the body temperature inside the human cells or the human body, right? So it should be 36 to 37 degrees. So for insects, it's 27 to 30. And then for avian cells, 38.5, a little bit higher. And then for cold-blooded, of course, it should be lower temperature, 15 to 26 degrees Celsius. And then osmolarity would be also another requirement, because osmotic pressure of the medium is critical for the growth as well. So it affects the temperature and even yung atmospheric environment nila. So cells are affected by that change of positive or negative osmolarity pressure. So larger molecules in the medium contribute little to the osmotic pressure as well. Okay, so just a figure just showing how it affects, like the pH affects the cell culture. So usually we use sodium bicarbonate or simply bicarbonate to somehow optimize the pH of the cell. So we put either like 1% of sodium bicarbonate, but it depends on the cell that you will be used. So here the figure just shows that the growth of the cells is different, or of the cells at different bicarbonate levels. So these are the concentration of bicarbonate level. So if it's usually higher, so the increased bicarbonate levels will also raise the pH as well. So another one is, of course, temperature. So just like any other models or systems. So the temperature on this one is a CHO cells in the growth and then the recombinant protein yield here in vitro. So the cells were counted and the secreted level of protein were measured after 4 days and at this indicated figure. So it increased at the level also at 37 degrees Celsius, if you cannot see here. Here. So somehow you can, it is also affected by temperature and also this increasing osmolarity pressure or osmolarity pressure. So the effect of this increasing osmolarity on cell is also differs from different concentrations. So here, as you can see, the optimum would be at until this point. So this one is almost 300, which is around approximately 290 OSM or micro OSM. Okay, so now let's move on. Oh, am I too fast? Anyways, later on, you will be having this PPT presentation or reading slide, so you can review it further later if I'm very fast. So another requirement would also be or things to consider are the extracellular matrices. So the effect of different medium volume on different cell number as well. So it's like also like imagine if you are in the level, of course, the oxygen level will be different if the level of medium is too high. So too much medium in the flask or plate results in increased depth of medium over the cell. So that means there will be a decreased oxygen. So it will not be good for the cell. So this is frequency result to the decreased cell growth or even yung population doubling niya and then yung function niya. Even like here, you can see it has decreases protein levels as well or protein secretions. And then here you can see the effect of extracellular matrix or the extracellular environment. So here they use one cells. So the primary culture here is from the rat, like the root ganglia sa brain. So it was grown in two different plates. So one here is laminin. So it's somehow coated with laminin, and then this one is polylysine, and this one's fibronectin, which is also a good one, and then collagen coated plate. So they have different coatings. So each plate, so these are adherent cells. So they adhere to a surface. So just like inside our body, so the cells adhere to something. So this extracellular matrix allows them to grow more. So the effects of this will also depend on which surface material they have. So here we have different type of membrane materials. So we have PET, PEFT, polyester, PC. So here, depending on what type of cell you have or also what type of experiment you will do. So you should also take note, like for example, if it's tissue culture treated or TC treated. So this one usually is the one that we use like normally in the lab, traditional one. And then others could also be used, but it should not be, or it's for a different type of special experiment. Okay, so for example, if you're doing immunohistochemistry or transport experiment, so you do this PET or you use this PET na plastic na coated or parang low pore density material. Also, like other experiments as well, like for diffusion experiment that requires matrix, but usually this one, we use this one. Okay, so various types of membranes and configuration can be used in different cell substrate in vitro as well. So again, let's move on with the media requirements and supplements. So the cells require, of course, sterile environment or clean environment. Sometimes, especially here in the Philippines, it's really hard to have a completely sterile environment. But as much as possible, it should be clean and has filter, or you do it inside the the hood, okay? And a supply of the nutrients for the growth. So of course, what is usually needed, like media that is needed in cell culture, we have the basal media, and then the complex media, or basal media usually are are composed of amino acids, vitamins, fatty acids, and lipids. And then other types also, they have this different complex media for different, for example, cells. Like primary cells are quite to challenging to culture. So there are special type of media. Okay, so maybe you can also actually make your own formulation of media and then check if the cells are growing, especially for primary cells. And now, ang pinaka parang great field would also be the 3D culture. So which like method, like a scaffolding, what's that? Like a different um what's that, different matrix that you can use, like a matrigel, so that you can form a 3D layer, so something like that. So there's a lot of areas to explore nowadays. And then we also have like a serum free medium and a special medium for like insect cells. So we have PBS. Usually these are a balanced salt mediums. These are just usually used to wash, but it has important role in culturing the cells. You can use deep PBS as well, or HBSS. And then basal media are usually MEM or DMEM. So that's the traditional basic media. And then we have complex media for different types of cells. And then serum-free media used for serum-free applications as well. Okay, so as said earlier, so like for BME or something like DMEM as well, or Eagles basal medium. So they have amino acid, vitamins, inorganic salts, and others. So amino acids, they have different components as well. So here, so you can see it like when you check the container, you will see this one, especially if this one is always special mention if glutamine is present or not. And then we have the vitamins, inorganic salts, and others. So additional components can also be present in other form of media, or other types of media. So next is, of course, for the cellular environment. So these are just the important components. So serum is a complex mix of albumins, growth factors, and growth inhibitors. So usually these are from animal fetuses. So we have this fetal bovine serum. We have NCBS, or new calf, NCBS, new calf serum. Okay, so we have also inorganic salts, as said earlier, buffering systems such as the sodium bicarbonate that is mentioned earlier, and then carbohydrates as well. Of course, it's important, the glucose, the galactose, and the maltose and fructose. So of course, you have been teaching this maybe in your bio courses. So these are the application of how it is used by the cell. Okay, so amino acids, of course, building blocks ng protein. So the essential amino acids, usually we call it EAA. So sometimes there are supplements that are EAA. So if it's, for example, primary cells, it's hard to propagate. Sometimes you add supplements such as EAA or additional amino acids to the media. And then, of course, you have vitamins, proteins, fatty acid, and lipids, trace elements such as zinc, copper, and selenium. So usually this one, if you read some papers, there are a lot of applications or functions of selenium in the cells. And next is antibiotics. So it is not really required to put antibiotics, but especially here in the Philippines, we always add like 1% AA or anti or the antibiotics just to protect the cells. But sometimes there would be some issues maybe of the antibiotic resistance. So when it's contaminated, sometimes it's hard to revive. But of course, it's not really advisable to revive a contaminated cell. So if it's contaminated, of course, you need to throw it already. So preparation of media. So there are different types of media. You can buy it in powder form or also in um, ready-made form, like in the 500 ml container. So for the powder, you have to, of course, dilute it, and then in maybe 1 liter, usually 1 liter water, but make sure that the water is ultra pure water, so it doesn't have any ions or anything. And then it's what's that? It was autoclave as well. And then because it's from powder, you need to use some filters also to check, of course, there's no debris or anything in the cells. Okay. So let's see. So here, just the standard cell culture technique. So we will be doing this later virtually. So using lab simulation as well. Okay, so from thawing, so we'll be thawing cells and then subculture and then cryopreserving or freezing the cells using the Mr. Frosty. So here I just made just the flow, sample flow. Later on, you can review this, so but we will be doing this later. So from preparation, of course, of the materials, and then getting the samples, and then also, of course, resuspending or collecting the cells, resuspending it, and then plating it and putting it in their optimum temperature and environment inside the incubator or CO2 incubator. So take note, this is a CO2. We put it in a CO2 incubator with 37 degrees Celsius temperature and 5% CO2. So here it's the same. So this one is just thawing. So from the freezer or from the like liquid nitrogen to the culture. And here is from the culture, and then we will split it. So here we call it like in different areas, we call it, depending on the lab, we sometimes used to call it passaging, or you can call it subculture, or simply you split one cell or one flask here, you split this flask, can use it. You split this into two or even four, or it depends on how many cells that you have got. And then it depends on the availability or characteristic of your cells as well, or your population doubling. So later we'll be doing this. Okay, are you excited? And then of course freezing. It's very important to have a stock or a solution, or like a stock or a backup of every cells, especially the low passage one. So if things didn't turn out, you have to get, of course, another set. So it's good to always have a backup. Okay, so of course, cryopreservation or the freezing. So the aim of this is to have a stock and store to prevent the need to have a cell, to buy another cell line again. And it's very challenging to buy here in the Philippines, especially mga cell lines. But nowadays, they have an official distributor like for ATCC, so it will be easier now. So cell preservation, so they use this cryoprotectant. So usually or traditionally, we use DMSO or even glycerol. So it may be added to the cell culture media. Sometimes we add additional serum, so that the cells are really protected. And then the DMSO is the one that reduces the ice crystal. But there has been issues nowadays on the toxicity of DMSO or the effects of DMSO. Okay. So counting of cells. So maybe some of you already have experience using a hemocytometer, right? So we use this even micro, right? So here we use trypan blue. So later in the afternoon, we'll be discussing all this also in detail. So how to count cells here. So how to also compute the number of cells. So calculate how many cell density is. So usually you have 4 areas here. So we count all that, but we just count there's a rule also accounting on the sides. Is it here? So here, so you should count only this part. So the insides, if it touched this part, like here, yung mga part na ito, if it touch, you should not include it in the count. Okay. So anyway, you can somehow count it by using the average of the count here, here, here. You divide it by 4, and then you times it by the dilution factor, which is, for example, this one, we use 10 microlit of trypan blue and then 10 microlit of your sample or cells. So that's the dilution factor 2, and then times 10 to the 4th, or 10,000. So that's your total density. And then you compute the cell viability using the average live cells over the average total number of cells. So nowadays, there are automated cell counter already. So, but it's good to know the traditional methods, because sometimes sometimes they go haywire, sometimes it's not working well. So it's good that you know the traditional way of counting, so that worst comes to worst, or like emergency purposes, you know how to count the cells. So here, counting, I also used hemocytometer to count liver cells from the mouse. Okay. So, and check also the viability. So nowadays, you can just put it in a machine and then it usually counts. But sometimes I don't trust some of the machines. So it depends on the machine and also the condition of the machine or how old it is. Okay, so next is cell-based assay. So after, of course, that's just the basic somehow techniques. But then what are the downstream assays? So what will we going to do? What are the research questions that are going to answer? Okay, so it depends on the problem that you have or the research problem that you have. But most commonly here, we have like cytotoxicity assays. We check different, we plate different cells. So from here, from the flash that we have done, we plate it in a 96 well plate and then treat it, determining also the cytotoxicity using LC50 or IC50. So yeah, usually here in the Philippines, they used MTT because it's very cheap and efficient, but there are a lot of ways. Like for example, here there's a luciferase assay. So they have dual firefly luciferase reagent, but they use what's that, luminometer. Okay, so they measure the luminescence here. So and also like most it's less toxic to the cells, and you can see the real-time viability as well. So this one, if you read it once and then you already check or you already put the MTT here, so you cannot use that cells anymore. Okay, so it's not safe for the cells. So it's like an endpoint assay. Okay, and then there's a lot more that maybe we can discuss later on in the afternoon. So next is contamination. So of course, it's the nightmare of every cell culture or biologist, so contamination. So even it can be contamination from bacteria or even some viruses or even some mycoplasma, fungi, or something like that. But we can also have a cross-contamination if you're handling like different types of cells. So sometimes you have like a lot of plate, and then you get confused of which is which that you are going to deal with. So anyways, it's very important, and you cannot ignore if you found some contamination. So the most, of course, the most practical thing to do is just to throw it, okay, and then get a new set of lower passage cells and continue with your experiment. Okay, so let's move on. So next is this one. So cell lines versus primary cells. So of course, cell lines are, as I mentioned earlier, so these are permanently established cultured already. They have been there is a high mutation and modification rate, especially in high passages, and then not as physiologically relevant. So we now usually shift to primary cells because it's from the primary or from the tissue, directly taken from the tissue. So somehow they are closely, the physiological function is closely relevant, and it's a stepping stone in vivo model or like mouse model or other things like that. Okay, so maybe I'll make it faster lang. So maybe primary cells application, we have it here. So cellular pathway analysis, therapeutic strategies, gene profiling, high throughput analysis as well, comparative studies, metabolic disease research, adult stem cell research, regenerative medicine research as well. So these are just sample primary cells that I have got it from the primary cell lines in PGH, so for breast cancer, from breast cancer patients. So 3D culture, now it's most closely to the clinical part. So it's very, the advantages are it's very optimal simulation in vivo conditions with minimal genetic drift in long-term culture. Okay, and then, but disadvantage, it's a little bit hard to, there's a need of, very challenging, there's a need of expertise really to generate such organoid spheroids or mammospheres as well. So here it's just a comparison of organoids and spheres. So organoids here, so basically organoids is the one that are treated or cultured in a plate here, like with a matrigel. So spheroids are formed in the cell lines or tumor biopsies as well. So they're really are floating cell aggregates. So sometimes you need like a U-shape special what's that, plate, so that it will grow well. Okay, so we have here the comparisons of animal models, 2D models, and also 3D models. So here, of course, 3D model, somehow it's in between. It transiently resembles the cell organization, as well, of course, as the in vivo culture. But somehow it lacks potency or self-renewal and differentiation. Okay, this is just a sample cells. I think, yeah, I think it's MCF or breast cells. I just treated it with endocrine disrupting chemicals such as BPA. So it this was analyzed in a high throughput machine and a fluorescent machine. So it's somehow very comprehensive machine because it's a high throughput, and it can measure different types of functions or different types of parameters as well. So you can check a lot of physiological activities. And then next, here in the 3D culture, as you can see, so it's heterogeneity, it's different. So of course, it mimics closely what is inside the cell. So everything that the proteins, the growth factors in the extracellular matrix and the cellular signaling can be mimicked by the 3D culture, as well as their structure as well. So they have different types of cells. They have the cancer cells, of course. Sometimes it is in the original environment, it's co-cultured with fibroblasts and immune cells, etc., etc. So it's very comparative with the inside the body, with the clinical model as well. Okay, so okay, let me know what time is it. Okay, so the cell as a application in its industry. So the cells expressing commercial settings. So for example, here it can, some of the, like for example, the media or the cell that you will get, for example, in the primary or in the primary culture, and then you somehow develop it into immortalized version, or you do a lot of maybe H-tert or transfection, or um, checking or maybe differentiating the cells or developing into a new stable cell, somehow you can patent that type of cell, and then you can name it with your own name or with your cell. So it's patentable, and then of course, cell line as an industry, you can bank it as a repository, and then somehow distribute it with the proper legal process. Okay. So for example, here the cell bank is of the production of cell line for recombinant commercial products. So just like what ATCC is, or other cell repository as well. So there's a lot of industry or commercial applications to cell biology as well. So not only that. So just to mention some of the problems and limitations. So we have the source of the cell. So sometimes it lost its differentiated properties. And it's very difficult to relate the cultured cells to the functional cells in the tissue from which they are derived. So it's still better to get a primary cells if you're really dealing with, for example, disease model of some sort. So sometimes you need surface cell surface markers to do such experiments to know which the source of the cell is. Okay, and then instability of some cells. For example, cell lines. Like I remembered, I have managed like a cancer cell line, I think MCF, until like 100 plus passage. So they have gone unto up till to that passage. So imagine how many times they have split it, and then but they still grow. Okay, so but maybe the characteristic is just different because they have been exposed to a lot of stress and like culturing and then centrifuging, trypsinization, and all that. Next is cost and expertise, so skill in handling. So there's a strict adherence in aseptic requirements. So it's very, very strict. So you have to wear your gloves, your PPE and everything. And then also you need to consider microbial and chemical contamination as well, cross-contamination, as I mentioned earlier, mislabeling and switching of the samples. So have to take note of that, and also the differentiation of the cell. So where the cell loses its differentiated phenotype, and then somehow it can also happen to have a reverse differentiation. So that kind of things happen. So it can be caused by overgrowth of undifferentiated cell or the use of other like selective media. So you have to take note of that. Actually, like some of the experiments are simple, but then you need to be very somehow meticulous on it. Okay, so now why are we studying cell biology? So why cell biology is important? So anyone, maybe later you can put it in the what's that? In the I can read it later. We can put it in the chat box. So maybe have you ever feel ill, or maybe you felt you had contacted COVID maybe, or sana hindi naman, and then even if like a simple like diarrhea or tummy bug, so it has been in your cells that were affected by, for example, like food poisoning or toxins from bacteria from a bad food. So you may know if someone who has been ill or diseased, of course, a lot of maybe all of us, like for example, someone you know has meningitis or dengue, what else? Malaria, diabetes, or maybe some types of cancer. So even if no matter what stages it is, or maybe AD or the Alzheimer's disease for our elderly loved ones. So all these diseases and disorders are caused by the problems at the cellular or even molecular level. So this physical damage, such as the burn or minsan wound or broken bone, so it can cause a damage at the cellular level or the cell level. So by understanding how these cells work inside our body, so in healthy and disease states, so of course we always have a control, like it's a normal state and then disease state. So it's not just like the effect, but also the mechanisms, the reason behind it. So the cell biology is working in the animal, plant, and even the medical sciences will be able to develop new vaccines, especially now, right? So you can use, of course, viruses cannot really survive on its own. So viruses depend on their host. So which is like for example sa COVID, which is like for the yung cells natin sa lungs, or for the in vitro environments we use vero cells. Okay, so yeah, things like that. So more effective medicines, or maybe alternative medicines as well, or like extracts from a native plant, some things like that, or improve qualities also of the increased knowledge to better understanding how living things live. Okay, so eventually here it will be possible to produce a health somehow health forecast, or analyzing a database of the genetic and cell information. So that's what, so our we were still in the infancy in the Philippines of testing that kind of, we don't have that GWAS, or like what's that, like GWAS like in the US, but using like maybe starting here, maybe like being aware of the applications of cellular and molecular biology nowadays, and sharing it maybe to your loved ones, to your students, of course. Not only like that, of course, the theories, you can read it in the book, but then how will you use it in our life? So like in the research, okay? So using this, you will be able to take more control over, like for example, your health, of course, your application niya sa preventive medicine as well, not just about the disease. So maybe like for example sa human fertility or reproduction, o yung DNA testing natin, that's been archaeology to provide evidence in living person related to like a long dead ancestor, right? So that's what the applications are nowadays. So for in plant science naman, it has been used to show that two plants that look different may have the same genetic origins as well. So cell biology also has part to that. So another one is forensic medicine. So like for example, what happened like earlier this year, right? So uses that cell biology and fingerprinting or DNA fingerprinting to help serve like solving murder cases, assaults, and other cases as well. So um, that's why it's important. Cell biology is important para hindi maka-escape and may escape yung mga criminals natin. So of course, all that biotechnology is the one that we use as technique, right? And information from the biology. So they get the information from cell biology to genetically modify yung mga different, like for example, plants or crops to be more resistant. For example, sa mga drought or something like that, to ensure high quality of food is available to all. Okay, so at lower cost, and to produce purer medicine that are not toxic to our body, but um, also it's what else? So like also sa mga transplant, or sa mga patient who need transplant, right? So cell biology is about or all this, okay, and can make an important or exciting career also in the future if we have all the facilities. So now actually there are a lot of cell biologies that are Filipino cell biologies all over the world. But here in the country, we have some few, and it's just, I think, like 10 years. So I'm a member of a PSEB, Philippine Society of Cell Biology, and it has been found just 10 years just because of a simple like round table talk or like chikahan. But they just suddenly like, oh, maybe we should also start this type of network so that we'll be updated what cell biology is, like the current researches in cell biology. So it's important for everyone also to feel informed about the increase of knowledge on cell biology and how could it affect him or her, and the society as well. So the basic knowledge of cell biology, including yung genetics, which will be discussed later on, and then also it's a Saturday, and then tomorrow will be molecular biology as well. But you can freely explore papers in the internet on recent studies on this. Okay, so just a quick applications of cell biology. So here in medical research, of course, we have cancer research, drug resistance sensitivity tests, model systems, diagnosis as well, virology, gene therapy, genetic engineering, environmental toxicity. So these are all related to the applications in medical sciences, which are very the most obvious application that we know. But also, it is also the core of life sciences or biosciences. So cell biology is the core of the life sciences, such as this molecular biology, the structural and systems biology, biophysics, developmental biology. So it somehow it is the source. It has become the source of this core life sciences. Okay, and also next. Um, Cell biology also, it does not only cover the basic science or the medical science application, but also the applied science. So as mentioned earlier, medical science or the clinical sciences, pharmaceutical sciences, bioentrepreneurship, and even agriculture and sciences, as I have discussed earlier as well. So there are many, many more applications that may be not well discussed or not well dove into it or not well explored. So just as a conclusion, so life scientists want to understand how biological systems work, right? So these systems include yung molecules, yung cells, yung organism, and ecosystem as a whole that are very, very complex. So biological researches in the 21st century involves experiments that produce these huge amount of data. And of course, cell biology has a somehow good source of data. So how cell biologists can even begin to understand this data and how to predict how, of course, the systems work. Okay, so together with the life of the core or the core, what's that, biosciences, core life sciences. So cell bio is a great source of tool that can help solve the answer the global problems that bridge the gap in the biological researches here in the 21st century. So that's it. Okay, so cell culture, I'll just end it with this quote that cell culture sa mga plantita dyan is like a gardening. So you sit to it and you look at the cells and then you see someday and say, you'll know that it doesn't look right. So somehow it's also like your baby that you see it grow and multiply. Okay, that's it for the first session. So maybe we can now go on to our questions.
So thank you once again, Ma'am EJ, Professor Saho, for that very wonderful lecture on introducing, or rather for introducing us to important technical terms as well as techniques in the field of cell biology and how basically it is intertwined with other biological disciplines. So thank you once again, ma'am. So thank you also to our participants for participating in our chat box, or rather in our chat feature. So buhay na buhay din po tayo kahit sa chat feature. So if or during our break, so maybe we can feast our eyes on some insights from other participants as well. So before we proceed with our demo proper po, we are going to have first our 10-minute break. Okay, so for our merienda or CR break. So see you later.
Naririnig ba? Ano na siya? Happened. Tapos nag-i-stop.
Baka hindi maayos yung pagka-download. Marinig.
Dito rin ako nagsisimula.
Okay, so hi. Hi, and we are back na po. So for the demo proper po, titled Demo of Cell Culture, Plating, Split, and Freeze Human Cell. May I now turn over the platform to Professor Saho from EG.
Okay, so I have read your takeaway. So I'm glad that you have learned a lot somehow or realized a lot sa presentation earlier. So for the introduction. So now maybe let's move on to the demo or virtual lab simulation. Okay, so let me share my screen. So did you have a good break? So may I know, so I think most of you are high school teachers, right? And then some I saw we have here, I think college level faculty as well. So anyway, so here, can you see my screen already? Okay. So
Yes, we
hopefully you can see my screen. So here today we'll be using this Labster simulation. So most of the um, lessons here and modules are um, here are from like different sciences background. So we have like general biology, microbiology, chemistry, ecology, etc., etc. So there's a lot of levels, and then we also have like high school level, and then this one mostly are what to call this? It's already somehow college level, but it's still basic that I think even high school level can understand or teach. So here, so this is Labster lab simulation. So here they have a module that includes simulation and also like a lab manual. So which has like the objectives, and then here you can see there's a theory pages, and then here you have you can see it's 61 minutes. So hopefully since we already tackled this earlier, so in the lecture part, so now I hope this will quicken our lab simulation. So today we'll be doing this. So can you hear the background music?
Hi there. Welcome to the Cell Culture Basic Simulation. In this simulation, you will learn basic procedures such as the aseptic
I'll
technique,
just
cell passage.
turn off the voiceover and I'll be your voiceover for today. So this is how it looked like. So you can also use this as a teaching tool for your students. So you know the kids nowadays, they have a short short attention span. Okay, so this one, like there's a study by Nature journal that gamification of a module is an effective way of somehow getting the attention of the students nowadays. Of course, like now we're shifting into the new normal, so new platform and all that. So this one actually it's not for free. So if you wanted me to hook you up with a Labster representative, si Shahira, so she's from Denmark, so she can somehow maybe give you free access for a week. So maybe you can talk to her and maybe talk to your institute as well if they are interested for this kind of online platform. But then again, this is an outsource, but maybe if we can, like in the Philippines, we can do some kind of like this app that would gamify techniques that can be used in teaching, like a new pedagogy, also like somehow it can we can adapt to this new platform or normal in this new social in this new distant learning that we have. So here we have the cell culture basic simulation. So can you hear me well, or should I remove the music? Okay, I think better without the music, right? So you will learn the basic procedure, aseptic technique, cell passaging, and cryopreservation. Okay. So here, I already know how to do this, so maybe I'll just guide you. So here, there are quizzes that can be that will be flashed. Maybe if you can flash your answer in the chat box, so I can also check if I have time. So maybe I'll check the chat box. Okay. So maybe you can answer with me, okay? So we have a virtual assistant here. So now we're going to culture some skin cells, okay? So their lab here is studying cancer research, or specifically skin cancer, and then trying to develop treatment for it. So we'll be culturing fibroblasts, or the skin cells that are and then we'll be massaging them and also freezing them. So just like what we discussed earlier. Okay, so first things first, of course, PPE, or protective equipment. You need the lab coat. Okay, so now we have the lab coat, and now we'll find the gloves. Okay, so we'll get the gloves somewhere here. So you can see the arrow. Just follow it. Follow the arrow. So as discussed earlier, cell culture is when scientists remove the cells from an organism and grow them in an artificial environment, often in a petri dish or other container in the lab. So this allowed the scientists to perform experiments in a controlled environment. Okay. So let's check. So okay. So question number 1. So I'll be checking the chat. What's this? The chat box. Okay. So what is the advantage of using cell culture? So is it A? Is it because it's easy and highly cost-effective tool to study life sciences? Or is it B, you don't need specific equipment to do this? Or C, is it an easy but expensive tool to study different aspects of biology? Or D, or you don't need training for this? Okay, I see mostly A. Okay, let's see. Of course, very good. So you got it. So of course, we already discussed this earlier, right? So because like other choices are not also suitable as well. So there are many applications that we have discussed earlier, such as producing recombinant proteins also that can be also used for molecular experiments as well. So why is cell culture referred to as in vitro? So earlier we discussed what in vitro versus in vivo, and also we discussed like the primary culture and 3D culture. So what is in vitro? So is it because cells are not touching each other, or is it a process performed outside a living organism, or cells that have lost some of their proteins, or because changes occurs in the cell membrane? Okay, there's some answering A or B. Okay, so of course, I think most of you are correct naman. It's B. So in vitro refers to the study of cells or biomolecules outside the normal biological context. So it is in a controlled environment, so like in the flask or in the disc. So for adherent cells, what does adhesion provide? So this one's a tricky one. So ano yung we have what's that? We have adherent cells, right? So the one that sticks to the bottom of the plate. So why does cells adhere, or what does this adhesion provide? Does it provide signal to grow and divide, or to enhance survival, or signal to transduce different station signals? Okay, so there's some A, B, C, C. Okay, let's see some more. Okay, so the right answer is, of course, all of them. Okay, so adhesion, it's very important, especially for the adherent cells. So it initiates the signaling cascades that are important. So it's the main reason that we use the coated flask, right? So the one with the collagen, the one we discussed earlier. So with different materials in the plastic adhering. So okay, so let's start. We're now ready to do some hands-on work. So your mission here is to prepare first your reagent. So as you can see earlier in the schematic diagram that I made, right? So first step, first things first, let's get the things ready, or let's prepare the materials. Okay, how do we get back? Okay, so let's go to the freezer. So what can we see in the freezer? It's okay to interact. You can, so that I know, you can chat. So here inside the freezer, let's see, let's take a look. So here we have, this is what we call our basal medium. So we have here RPMI. So if you remember, this is the special, like somehow it says special type. So these usually are used for suspension cells, so not the adherent cells. So for example, lymphocytes. So usually this one I use for like splenocytes. So the cells in the spleens. And then most of the time it also is good for primary cells. So it looks pink or colorless depending if it contains phenol red or with no phenol red. Okay, let's see what's next. So here we also have what we call conditioned medium. So what is a conditioned medium? So this is a special type of medium. So recently it has been discovered that the cells like that grow in the medium for a long time, they produce something, like they produce growth factors that cannot be produced elsewhere. So this is made from any used media. So for example, you use it already for your culture. And it has a nutritional value, usually be in contact with the cells, and many applications as well. For example, for cell differentiation, induction, para ma-activate siya, and the delivery of engineered viruses and even therapeutic agents. So usually for stem cell experiments or researches, and this one, I use this for special cases, like for example, sometimes when I freeze the cell, it's better if I use conditioned media because there's a lot of, because originally I use more serum, and then more serum, and then the freezing media, like the DMSO, and then the media as well. But this one you can also use for freezing. Tried and tested. So next is the traditional one, or DMEM, also called the Dulbecco's Modified Eagles Medium, and it's one of the most used culture medium to grow adherent cells. So basically anything that not naman anything, but mostly like any cells can grow in DMEM. Okay, but of course with serum and other growth factors or supplements as well. So this is the basic component. Besides its basic components, it has phenol red. That's why it's red in color. That acts as a pH indicator. So sometimes if it gets like pinkish or maybe it becomes yellowish, it like the flask, it tells you need to maybe change the media already. And also it protects media from light or damage of light. Then lastly, we have here HPSC medium, so usually for stem cells as well. So it's specially formulated for pluripotent stem cells for the growth of these stem cells, and provides essential nutrients that keep their pluripotency intact, and it contains phenol red as well. Okay, so as you can see, this one doesn't have phenol red. So there are media that has phenol red and without phenol red. So sometimes, because phenol red has also an effect, and if you're studying, for example, what's that? Like for example, in EDC. So for us, we study endometrial, EDC, endocrine disrupting chemicals. So sometimes it affects your results. So it's better to use the one without the phenol. But just make sure your pH is always right. So here we have the serum. So again, let's let's get the medium that we're going to use today. So what is the medium that we're going to use to culture fibroblasts? Anyone? Okay, so of course, fibroblasts, we use DMEM, so the traditional way. So we got it right. So we'll put it already in the water bath. So we prepare the sample. So here, we put it here. Okay, and next, we'll also get, okay, what are the main components of the basal medium again? What is the main components of the basal medium? Is it the trace elements, or is it all of these one? Trace elements, amino acid, water soluble vitamins, or is it amino acids only, or is it water soluble vitamins only? Okay, so I think everyone knows it's B. So it's very, very clear that we have studied this earlier. How about L-glutamine? So this one, usually it's indicated in the bottle. You can clearly see this. So why is L-glutamine an important amino acid in cell culture? Do you know why? Is it the new one na? Okay, so is it because it's an amino acid or amino donor, or it's amino acid, or it gets so much energy from its anabolism, or it gets so much energy from catabolism? So are you familiar with anabolism and catabolism? Okay, so later actually we will discuss this in the metabolic activity in the cell techniques. So actually parang walang nakakuha ng right answer. Okay, so the right answer is L-glutamine is catabolized into the compound directly, so involved in the Krebs cycle. Remember that. Okay, so it helps the cells produce most of the energy they need to keep growing. Okay, so it's letter D. Okay, so what is the function again of phenol red? Okay, this is very easy. I mentioned this earlier. Okay, so does it indicate pH and protect light? Does it help the cell membrane stay permeable, or does it provide the source energy to the cells, or activate the signaling pathway in cells? Okay, so parang wala namang mali. I think everyone's right. Okay, of course it's letter A. So here we can see here the color. So usually here we have 7 point. Here when we newly put it in the media, maybe 7.4, and then if it goes if it goes to 6, it becomes a little bit yellowish. So if you see your flask, this color, actually like this color pa lang, you can see maybe 6.9. You changed the media already. Okay, so usually after 2 days you change the media. So cells grow well in 7.4. So make sure na ganito yung color niya. So 7.4 yung ideal color. Okay, so phenol red indicates pH, and it turns yellow when pH is around 6.8, and it turns pink when it's around 8 in pH. So just to keep track of your cells. Now, so when you are using non-phenol or no phenol red media, you should know your cells by heart. Okay, so of course you should know when you should change your media. You should know the population growth, or you just know it's like a baby. You need to know when to change the diapers, when they are needed to be fed. Okay. 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 It's okay if the power supply, if there's no power supply in your area, just you can watch maybe this later in YouTube. And then next is, what is the conditioned media useful for? Is it used to add is it added to the thawed cell, like after you get from the nitrogen tank, or is it only useful for the cell type used to make that conditioned media? Or is it useful or helping grow very low density animal cells? Or does it elicit cellular responses? Okay, I see different answers, B, C, B, C, B, C, D. Let's see, ano ba ang tamang samples? Ay, ang answer. I'll check. I think it's letter D. I'm not sure also. Okay, it is correct. So conditioned media can help cells grow a bit faster. So besides providing yung basic requirements that we discussed earlier, there are other growth factors as well na present in the conditioned media. And then therefore it can grow the cells and grow slower than needed. Okay? So next, what are the optimal conditions to store the cell culture media? The earlier, the media that we have. Okay, is it, let's see. Are you starting answering already? So is it at 37 degrees Celsius in the dark? Should it always be inside the laminar hood? Should it be at 4 degrees in the dark, or should it be in the workbench out of direct sunlight? I think most of you are correct. Okay, so sometimes, so since yung ref, I'm not sure if yung ref nyo sa lab, usually when you close it, diba, the light is closed naman. And then if you open it regularly, maybe some of the media, better if you put a foil, especially when you already prepare it, and then you seal it with the I forgot again the term. So you can seal it with that thing to seal it. Okay, the plastic thing. I forgot the term anyways. So sometimes we need to add serum in the culture media. And most of the time this is the case. So serum such as FBS or fetal bovine serum is usually comes from yun nga, animal fetuses, and then has high concentration of growth factors and hormones that stimulate cell to grow and provide. So let's get the serum in the ref. So you see here, originally it's in a 500 ml container. So when we receive it, as soon as possible, we heat inactivate it, and also it depends on the type of your serum or the brand of your sera. And then this one, you alico it to 50 ml. So let's get one so that when you use it, you don't need to yung freeze to. So parang, I don't know if you're doing this at home then. So yung mga chicken. So I usually separate it so that I can thaw only what is needed, right? So just like at home, right? Which one of the important components of the serum? Anyone? Okay. So is it minerals, amino acid, vitamins, or growth factors? Okay, wala pa namang mali. Okay, very good. Okay. So it's letter D, of course, growth factors. So serum provides growth factors to increase the growth of the cell. And the next is, what's the best way to store the serum? So I'm not sure if you can see yung division sa ref nila. But here, is it covered from light? Is it in a big bottle? Is it always in the fridge, or is it in alicos frozen in -20 or sa non-frost freezer natin? Okay, very good. Very good. Tama kayo. So wow, yeah, you're all correct. I think wala namang mali. So the best way, of course, is in alicos, as I mentioned. So to avoid yung freeze-thaw cycles. So you should also do it sa mga, let's say, what's that? Mga giniling nyo or some food in your freezer, so to avoid freeze-thaw then. So why do we pre-warm the reagents that we need to use at 37 degrees Celsius? Is it A, to induce the thermal shock to the cells, B, to inhibit some of their components, C, to activate their components, or D, to avoid thermal shock to the cells? Okay, I see some C, D, C. Let's see. Okay, medyo hati. But the real answer, or correct answer is, of course, to avoid of course, like the cells, the temperature should be just like the temperature inside the body. So to check if the temperature is right, so usually it depends. So for example, you have yung serums which is frozen, so it usually takes a long time, maybe 30 minutes, like here or so. But you can check using your hand, so because you have a glove, so you can check using, I'm not sure if you can see me, but you can check with your like pulse area, and then the temperature should be like parang sinisinat. 10 minutes left. Okay. Okay, so anyway, so it's like that. So let's maybe fast things up. Okay, so after that, so since it's virtual naman, so it's done already. So let's open. So I think even though we cannot finish din naman, so this is just a demo, and then just to show you some of the basic techniques. This one you can I can send you a video of this, so that maybe you can review. I made already a pre-made video. So okay. So first, let's open the hood here. Let's open the hood. So usually, there's a lot of steps pa. Like for example, you turn on the UV for 15 minutes and you disinfect the area. So the airflow, maybe you have seen one of this. So it is a hood that protects the working environment from dust or airborne contaminants. So I'll just make this a little bit fast. So of course, everything that you put inside, you always aseptically clean. Okay. So for me, I usually use tissue. What happened? Okay, tissue, and then spray it with the alcohol, and then I spread it on the materials I need to put inside the bench. Okay, I already sprayed it, and now we'll okay, spray also the serum. So the instruction is seen here naman. So let's put the serum and medium inside. Oh, here. Okay. And then also the serum. Okay. So next, what atmosphere, what is the atmosphere inside the airflow hood like? Okay. Is it very clean but not absolutely sterile? Is it full of CO2? Is it lower in oxygen than the air outside? Is it absolutely sterile? What do you think? Oh, are you sure? Wow. You're all okay, okay. I think yeah, meron some of them. So it's not actually absolutely sterile. So it has HEPA filter. It filters some of the like debris, bacteria, viruses, etc., etc. But then it's not completely sterile because somehow it just filters. So it looks like this. I'm not sure if you have. So there's a hole here. So the airflow here, and also it flows here, and then go down here. So when you the cap, so there's a argument with this. So you should you put the cap down or up? Of course, as much as possible, if you can hold it with your hands, even though if your hands are not all preoccupied, you can hold it, or you can put it sidewards. Okay, so that because there's a lot of argument, like even in micro, like it should be up or down, because like the air will flow down, but also the what's that? The surface is also not that clean or not completely sterile, right? So you can put it sidewards if you can. Okay, next is how much the sash is the adjustable transparent cover of the airflow or air hood will be set. Okay, so is it up high at for the ease of the use, so depending on you, or is it at the comfortable location of the user, or is it at the indicated level when you use it, or at the indicated level when you're not in use? Okay, we have letter B, we have letter C. So remember, okay, so there's a level there, that there's an indicated level, but you always just put it when it's in use. So you, of course, so that you can work on it, right? So letter C. So let's see. So which rule you should follow when working with animal cells on an air flow hood? So is A, never open any item outside the hood, or you should not, you should, or you don't need to worry about aseptic technique, or you can't stop working until they finish with everything inside the hood, or use quick movements inside the hood. Okay. Let's see A. There's a lot of A. Let's see nga. Okay. Okay, so perfect. We can now somehow like start working on the real laboratory. So now let's rinse the culture flask. So we will prepare also with the glasswares or the plastic wares instead. So spray. Oops. Wait, I can't press that alcohol. Okay. I think I sprayed too much. Anyway, so we'll put it inside here. So what is the main characteristics? Okay, we have 5 minutes left. Maybe we'll just, this is just a demo, so I can just, let's see what part we can do for now, and then the rest would be sent to you via video na lang. So what is the main characteristics of the vessel specifically designed for culturing adherent cells such as fibroblasts? So the surface is specially coated, or is it because the shape, the size, or uncoated? Okay, sige. So very good. I think everyone's correct. Okay, so we discussed it a lot of times earlier. So, okay, now let's get 50 ml. So this is a pipetor. Then this is a 50 ml pipette. So we get we get media here. So this is 500 ml. So we will throw this. We'll discard 50 ml. But actually, it's very sayang. So you can still store it in another, like here in the Philippines. Of course, we store it. Like even other countries, you still use it. You store it in a new 50 ml. So we have removed it because we have removed 50 ml because we will add 50 ml of FPS to the media. So it's 10% FPS. So complete medium with 10% FPS. So why is now it's called complete medium? So now we have a complete medium, the DMEM with FPS. So is it because it supplies all components of the cells needed to grow? It facilitates detachment of the adherent cells, or is it necessarily only for some animal cells, or it has been contact with other cells? Okay, we have A, we have B. Okay, but the correct answer is, of course, the majority. Okay, it's A. Of course, we discussed this earlier, right? So we add serum that contains growth factors and hormones, and also the basal medium who has the vitamins, the trace minerals, and amino acids. Okay, so does a complete medium need to be placed at -20? So do you think we need to place it at -20, just like the serum? Is it yes? Many biologicals last a long time when you store in -20. Or yes, the serum comes from a vendor frozen. Okay, and or no, it's -80 is a shock to work better, or no, its basal medium should not be frozen. Okay. Okay, parang dalawa pa lang nakikita. Okay, sige. Of course, it should not put it in the freezer, okay? So, once you already mix it, you store it at 4 degrees and for no more than 2 to 4 weeks to ensure all components are still active and avoid precipitation. Okay, I think we don't have enough time, but anyways, let's see if we can just finish like the preparation part. So can we add antibiotics to our complete medium? Is it A, never, or B, is it only when necessary, or C, always, or D, only during a cross-contamination? Okay, so very good. So only when necessary. Okay, so unfortunately, I think we don't have enough time. Is it right? What's that? Noel, can we still continue ba, or should we take a break?
Ma'am, unfortunately, I think we don't have enough time po.
Okay. So
yes,
anyways,
sorry po,
this
ma'am.
is just a demo lang naman. So we will be sending you a video on this so that you can also continue the whole process and you can watch it on your own. Okay? Sige. So what's the next part? Is it okay if I can have a very quick break
Oh, yes, yes, ma'am. Yes,
before we
ma'am.
have okay, sige. Okay, see
Ma'am,
you.
ma'am. Okay. So,
Yes.
ma'am, I think we can, ma'am, since it's already nearing 12 noon, ma'am, if it's okay, can we have our open forum, ma'am, for the Q&A session, ma'am?
Okay. Can we have like 2 minutes?
Oh, okay lang po, ma'am.
Okay,
Sige po, ma'am.
thank you.
Okay, so once again, thank you, ma'am, Professor Saho, for introducing to us into this very interactive simulation. So to our participants, I hope we have all bundled up or gathered up all of our questions from lecture 1 to the demo proper, as we will now proceed, as I have said, to our open forum po to answer some of your questions. So to our Zoom participants, may we please ask you to simply type your questions in the chat feature of our Zoom meeting, whereas for those viewing this live via YouTube, to type in their questions in the comment section. Okay, so again, we may type in our questions po in the chat feature for Zoom participants. For those viewing this live via YouTube, to type in their questions in the comment section. So while waiting, may we please remind again our participants that the evaluation form will be posted, or the link for our evaluation form will be posted later on or later this afternoon. So this evaluation form will truly help us to further improve in our upcoming webinars or workshops. Okay. Okay, so we have, okay, so marami pong nagtithank you. Okay. So questions. So I think to start off with our Q&A session. So ma'am, we have here a question from Kenneth Frias. Can you use this same cell culture techniques to protozoans like ciliates?
Okay, so I'm not really familiar with the techniques on ciliates, but technically, you just need to have a specific type of media, as what I can say, right? So I can comment. Like, I think basically you can culture it with a specific type of media. So maybe a selective media for ciliates. So, because we have other, like for example, certain environment or physical requirements also that is unique for ciliates. So you can check on that. Yeah, the protocol on that. So I haven't really looked into that, but it's possible. Like the techniques or somehow the methods would not be, I think, not be that different. So it's basically just making the same environment where, like an optimum environment where ciliates can grow and culture.
So thank you, ma'am. Ma'am, we have here another question from Jim Carlo Pula. Thank you, ma'am, for the interesting topic. Ask ko lang if magkano subscription for the Labster. Monthly or annually po ba?
Okay. Actually, I can forward you the maybe email. I'm not sure. Maybe I'll post na lang Shahira's email din sa Facebook group. So the representative um, here sa Philippines, they have it here. I just I have it in the email, but I'm not sure. Later, maybe I can pull it up. Give me a second to pull up my email. But it's usually not monthly. It's I think per semester or annually. And then the fee is the subscription is for student. Actually, it's free for the faculty, but the student can pay for it. So there's already they will be the one monitoring how to grade them or something like that. And yeah, so if you have more questions, maybe I can just hook you up with Shahira na lang, the representative sa Denmark or representative here sa Philippines. Yeah, you need to ask. The process would be it will be the institute or the administration who can decide if they will, of course, purchase. Okay. Actually, we don't have that in our university. I think UP Diliman has. What else? A lot of I think even high schools have this already here in the Philippines. So yeah, maybe you can, because they will, if you email them, they will give you a 1 week, what's that? 1 week trial so that you can check to see if it works for you or your students. Okay.
Thank you, ma'am. Thank you, ma'am, for that answer. So, while waiting for other questions, ma'am, I have a personal question po. Since it was introduced that cell biology po is really intertwined and interrelated with other biological disciplines in the educational sector po kasi lalo na po doon sa mga fresh graduates ng senior high po. Upon asking po kasi doon sa ating ibang freshmen sa UP Baguio na BS Bio students, I asked them po what field of biology were they less interested in, and most of them answered po cell biology, and most of their reasons were because the terms were really technical and that they were really afraid of taking up or studying cell biology due to its technicalities. Ganun po, ma'am. So with this po, ma'am, what tips can you give us po to somehow lessen their fear of cell biology, lalo na po sa senior high?
Okay. So since the background, we're not really exposed that much, especially here in the Philippines, to researches on cell biology, and maybe they're overwhelmed lang, or I'm not sure if they're why they are afraid, but don't worry, like you can I have trained a lot of interns, even high schools, college level, even graduate school level, and they are usually they're good with it. Like a week is okay. So it's actually more on practice, or like if you do it every day, so it will become a routine already. So it's not uh the methods or the techniques any techniques can be learned, right? So we should not be afraid, but yeah, just take maybe some diligence to really learn the technique. So don't be afraid also to make mistakes, to get contaminated or something like that. But yeah, it's part of course the process of learning. Okay.
Thank you so much, ma'am, for those tips. Maraming salamat po. Ayan, so ma'am, we have a question from the SINSM FB page from Maria Benita Eloreta. Just curious to know, or please enlighten us, if regeneration of cells is one application of stem cell therapy. What is your opinion on the cell of stem cell and taken orally, which some claim it as life-changing way to treat metabolic disease like diabetes?
Mhm. Okay, interesting question. So, yeah, somehow the regeneration or regeneration of cell is there's a crosstalk with the stem cell researches. But actually, I'm not into that, like orally taking it, and especially there has been claims, like for example, like plant stem cells or something like that. I'm not sure the mechanism behind or the concept behind that, but for me personally, it doesn't make sense for me. But like there are stem cell applications, but it's not like orally taken. It should be somehow be injected or something like it's it's more on the system, effects on the system. It's not just taking it orally. Yeah, I'm not yet sure about it. I haven't read more like detailed or explore on the mechanism behind the orally taken stem cells. Mhm.
So thank you, ma'am, for those answers. So again, ma'am, we have another question from the demo proper from Labster. I'm from Joanna Rose Panergo. I'm also interested po sa Labster, how to subscribe and how much po.
Okay. I haven't really checked my email, but um okay. So you can subscribe by contacting their representative. I will forward her email sa FB group, right? I'll just forward it there. So maybe I will also. So for developing countries like the Philippines, they have special prices for us. So we have the cheapest subscription plan that are available. So maybe I'm not sure if it's the same to all, but it's better if you can just connect with her. But yeah, I was thinking like this one is a good platform, but this is from Denmark or like made from Danish people. So maybe if we could make also our own. So calling all this is a call for all like maybe computer experts there or mga app developers. So maybe if we can make something that is because the techniques here in the Philippines is a little bit different from like developing countries. So sometimes for us, we're more meticulous, we're more careful, especially for contaminations because it's very humid here in the Philippines and microorganisms thrive thrive better here or they thrive more than like of course yung cells natin or other experiments. So yeah. So maybe I'll just post na lang later. So maybe you can remind me, Dane or Noel, to post the email of the representative.
Yes po, ma'am.
Yeah, I'll try.
Duly noted po, ma'am. Okay. So thank you, ma'am. Thank you so much po. So I think that's everything from our chat box po from the Zoom, from our Zoom meeting. So again, unfortunately, that's all the time we have for this morning's lectures as well as open forum. So thank you once again to our participants for raising your questions as well as your insights on the first lecture as well as on the demo proper. Okay. Now, since it's already, or rather we are nearing towards 12 p.m. or 12 noon, we will all be having now our lunch break. But before we have our lunch break, we have some reminders that we would like to reiterate to everyone. So to our Zoom participants, may we humbly ask again, our Zoom participants to not leave the Zoom meeting if possible. Kasi po baka mahirapan po tayong pumasok ulit. But kung hindi po talaga kaya, okay lang naman po. Just a word of caution though po na baka mahirapan lang po tayo sa pagbalik dito po sa ating Zoom session. Second, for the evaluation, once again, it will be posted this afternoon. And finally, we will all be back. We will resume at exactly 1:30 p.m. Okay, so once again, we will resume but at exactly 1:30 p.m. So thank you, Ma'am EJ. And let us all have a good lunch. Kain na po.
Wait.
Fine, sir.
Mag-leave yata muna ni. Ay, mas may mga
So hello, good afternoon once again everyone. So I hope we all had a good lunch. Okay, masarap po sana ang kain natin ngayon as we will now have our second lecture proper on the different model systems bioassay techniques that can be applied in your students' research, still from our lecturer, Professor Sao. So, Ma'am EJ or Professor Sao, the platform is yours na po.
Okay, good afternoon. So let me share my screen. Okay. Oh, wait. Oh, okay. So wait a minute. So I hope everyone had a good lunch, and I hope you had your after lunch coffee already, so that hindi tayo aantukin today. Okay. So maybe give me some minute to open. Okay, that's it. So let's see. PowerPoint. Is it this one? Screen sharing has failed to start. Okay. Maybe someone can help me. I'm not sure. Okay. So how about this one? Can you see the screen now?
Yes po, ma'am.
Okay. Sige, sige. Okay, so let me start. So I hope everyone is awake. So we are all full from our lunch. So now we will move on to different model systems that can be used for your research or for your students' research if you are a faculty or teacher. Okay, so I divided this lecture into 4 parts. So we have the cytotoxicity screening using brine shrimp and trypan blue, and also the second part will be the metabolic activity assay. So another model system. So here we have the MTT, so the cell-based downstream experiments. And the next is 2 models. So we have C. elegans model and Daniererio model. So actually there's a lot more, but for the sake of time, so I just cut it into 4. Okay. So let's start or begin with the cytotoxicity screening. So here, I'm not sure if you're familiar with the brine shrimp assay, and then another one is the trypan blue dye exclusive assay. So why do we need cytotoxicity screening? So of course, like in different fields or researches, so everything, for example, should be screened. So everything should be somehow initially screened so that we will check if it's safe for us to take in or for us to continue with the research part. Okay, so next is it okay? So assessing the cytotoxicity is important in identifying different candidates as well for drug development or like a new drug discovery or new product. So promising biological activity versus negligible cytotoxicity. So you have to weigh in the pros and cons. So will you consider its effect or its safety? So of course, number one, especially when we're dealing with, like for example now, vaccines. So first, all of it should be screened. Okay, so in vitro. So in the in vitro stage, in vivo stage, and then also in clinical stage as well. So cytotoxicity screening also gives insights to potential adverse effects of the substance and relevant cells before engaging to a more expensive studies, like for example, a mouse study or in vivo studies or even clinical studies as well. So that's how we work for like different drug discovery and even vaccine, right? So from in vitro to in vivo and then to clinical. So next, screening also identify the compounds or substance which can pose health risk to humans. So here for the first part, I'll just want to discuss 2 models or models for cytotoxicity. So one is in vivo assay. So we have this brine shrimp assay or lethality assay, or it's also called BSLA. So we'll be discussing that, and also an in vitro assay. The simplest is trypan glue exclusion assay. So these two are specifically picked because these are very feasible assays that can be done apparently maybe in your own institute. So at a lower cost, and also but you also have to consider the pros and cons of these different tests. So first, let's discuss brine shrimp or the Artemia salina. So I'm not sure if any one of you have already, or maybe you can raise your hand. I'm not sure. Let me check the chat box. Okay. Are you all familiar with the sea monkeys? I'm not sure. I think this is like a toy. Yeah. So we have lalaman natin kung sino yung nakaranas na nito or have experienced this type of toy. Is it a toy? Anyways, I just read about this, and like it's interesting, right? So this one before it has been used and somehow commercialized um, in the market. So brine shrimp or Artemia salina, any of several crustaceans that live in salty waters. So now you can somehow imagine the media that you can use. Of course, it's salty water, so the media or the environment should be also salty water. So these brine shrimp or sea monkeys, so they have tails remotely resemble like monkey's tail. That's why they are called the sea monkeys. So you can see this everywhere. It is sold in the pet shops, and then yeah, it like before. I'm not sure if it's still out in the market, but it is also sold as like a toy. So uh, somehow they don't actually resemble these creatures in the packaging. Okay, we still have that here pala. Okay, so maybe I'm not sure if some of you have used this in your research. So this is such a good model. So here's just a introduction or just a brief background of its life cycle. So from um, so usually brine shrimp are used in the laboratory for testing like toxicity as well as screening so of chemicals. So brine shrimp come in many colors as you can see. So maybe you have seen maybe white version, pink version, green version. So different colors are probably an effect of either yung diet nila or even yung environmental factors as well. So here you can see, so at their early stage, I'm not sure if you can see the arrow. At the early stage, so dormant cysts, so they're suspended in the gastrula, and then after that, embryos somehow inside resume development. So if conditions are favorable, they will eventually move to this nuclear cells. So it's nucleus in singular and nuclei in plural. So this nuclei feed on the yolk and then soon develops and feeds on the food from the environment. And then you can see here, they have only one eye at this stage. So after that, small adults, the first larval stage, they breed, they feed, they swim through their feet, and then the male and females are somehow they look alike, and then they grow up to this adult stage, or they have their distinct features that is distinctive between males and females. So males hold the females with claspers. I'm not sure if you can see the claspers. Okay, so there's a distinction, and then active nopili releases from broad sac if conditions are favorable, and otherwise release too as cyst. Okay, so next. Okay, so their odd behavior is they swim upside down, so due to the positive phototaxis. And they rise within the day and sink at night. So what are the uses of this brine shrimp? So first, they are very important in the local economy. So they live feeds for fish, Also an indicator of water pollution. So it can detect like fungal toxins, heavy metals as well, pesticides, environmental pollutants. So preliminary assessment of plant extracts for toxicity or bioactivity. So here's the principle behind the brine shrimp lethality assay. So there's the ability of to test this substance to kill the culture brine shrimps and therefore determine its lethality. So the hatch shrimp moves into the surface and directed to the attractions of light because of their chemo or photoresist or photolethality. So when they're affected by test substance or any samples that you have, so the larva stops swimming and sinks at the bottom, and then it turns opaque or colorless. Okay. Its advantages mainly as it does away with the continuous culture of the test organism. So the eggs or the brine shrimp eggs commercially available in pet shop as well, so it can easily be seen or can easily be found. So another one is, or a lot more actually. So it's easy to rear, very robust, and then sensitive indicator of species. It retains viability for years, so it's a good thing. So if properly stored in dry condition. It is also rapid, inexpensive, convenient bioassay that does not require a septic technique. Uh, so it's somehow feasible here in the Philippines. So you don't need a clean room or a special room for this. So the cost of conducting an assay, so let's say it's not that much so compared to other assays. So it may be used in conjunction with hatchability assay. So there's another assay using this brine shrimp as well. So, but for time's sake, we'll just focus on this lethality assay. Okay, it's moving. Okay, so just an overview of the schematic procedure. So this one, just prepare the media. So this one is the artificial seawater. So you can prepare this by mixing and rock salt. So 18.5 gram rock salt in 500 ml distilled water. So it can be easily done at home, right? And then you can add the 20 milligram of brine shrimp eggs in the medium that you have, the artificial salt water, and then incubate it at room temperature under a constant light, okay, and strong erosion. So you can do it maybe in your um, institute, or maybe you can have your own DIY garage lab, right? So you can easily set this up. Next is, so of course you prepare your treatment, and then of course your control, and then you add 10 nopili into each well of the treatments or the control. And then incubate it at 24 to 74 hours. So depending on your experimental design. So slightly cover the plate with the lid so that it won't get contaminated. So after the incubation, now you can count the live and dead nopili using dissecting microscope. So I'm not sure if you can use it your naked eye, but at least you can have dissecting microscope. So if you want to take a picture of your samples, you can also have the adapter. You can easily, yeah, just buy an adapter ng microscope to the cell phone. So it can be easily done, and you can buy it sa mga Shopee, Lazada, or something like that. And you can use it in your experiment as well. And then record your data, and then you graph your data using this percent mortality formula. So percent or number of dead nopili over the total nopili times 100. So as easy as that. So I'm sure like your high school students would be would not be afraid to try this. Okay, so next is this one is just a picture of the complete setup of the assay. Yeah, so that's it. So just a simple brief background so that we'll have more time for more assays. So next is this trypan blue dye exclusion test. So we already discussed this earlier. but maybe we'll discuss this more in detail. So the mechanism behind is they use azo dye. So it's used for dyeing cotton textiles. So this dye is called trypan. So because it has the ability to kill trypanosomes, or the if you recall to your parasitology class in sleeping sickness, African sleeping sickness. And then it's also called diamine blue or Niagara blue, or vital stain to identify between live and dead cells. So take note that this one, so most of the stain that we have that are available are cytotoxic. So once you already use this, you cannot use the samples, or basically you have a 1-point assay only. So this one is also a little bit a traditional manual way of counting. So as discussed earlier, so this is one of the most common methods of assessing cytotoxicity, or actually kahit monitoring or counting of cells or cell viability as well. So dye is negatively charged and impermeable to viable cells with intact membrane. So only those who are able to enter the membranes are the dead cells. So here, so they can enter because they are dead. So because the cell membrane are compromised already. So it binds to the intercellular proteins within the cell and stains the cell blue. So as you can see here, so the one with the blue, so cell suspension is mixed with 0.4% dye, or usually in cell culture, we do a 1:1 ratio. For example, 10 microlit of the cells and then 10 microlit of the trypan blue. So 2 dilution factor. So visually examined under the microscope, so you can routinely perform while counting the cells using the hemocytometer. And I think we already discussed that earlier. So this is other reported applications of type trypan blue. So it's a simple stain also to visualize bacteria and viability of amoeba, and also examine infection structures on plant-fungal interactions. And it can also stain the Cryptococcus yeast as well. So here, just the schematic procedure. Actually, we already know this already. So we plate the cells, we incubate it for 24 hours. We treat the cells with your chemical of choice or maybe extract of choice. And then after 24, 48, or 72 hours, you monitor it, and then you collect it and perform trypan test. So this can be done maybe just for a small number of tests or like small amount, but it's for high throughput, like 96 samples or triplicates, it will be very tedious and hard. So, but it's a simple method only. So this one, we discussed it earlier. Okay, so okay, so after removing or collecting the cells, so you collect the cells, resuspend it, and then you put 10 microlit of trypan blue and 10 microlit of sample. Actually, I don't, maybe is it 0.5? Anyways, 10 is to 10 or 1:1 ratio. And then load 10 microlit of the mixture in the hemocytometer. And then you can calculate the viable cells by dividing the total number of viable cells over the total number of cells times 100. And then, as I said, all stains are potentially hazardous substance. So be careful. So here, as you can see, so this one, the blue one, are all dead cells. Okay, so you count only the gray one here, and then you should not count the one that is inside. So just count the outer area. So exclude the one that's touching the middle line of the bottom and then the right side. So let's see, what are the pros and cons or advantage? So is the price okay for conducting this kind of experiment? So it says here, kaya ba ng budget? So price is okay if considering only trypan blue and plastic wares and hemocytometer. But then again, if your experiment requires a skilled personnel, cell culture equipment or facility, and other equipment, cells, and our agents, so you might be able to consider or you should consider that expenses or budget as well. So what can be done with your budget? Okay, so next is also related to cytotoxicity. Cytotoxicity is metabolic activity in the cells. So a while ago in our demo, there's a question there on the conditioned media, right? So let's review that. So of course, cellular metabolism, so a set of chemical reactions that occur in the living organism in order to maintain life. And then it involves a lot of sequence of complex sequences and controlled biochemical reaction known as metabolic pathways. And then we have a catabolism and anabolism. So catabolism, it's the breakdown of molecules to obtain energy, and then anabolism is the synthesis of all compounds needed by the cell. So what happened in the conditioned media, right? So the conditioned media produces energy from the cells that are in the medium. Okay, so it breaks down the molecules inside the cells to obtain more energy. So that's the mechanism behind the conditioned media, that it produces a lot of growth factors, maybe signaling surface proteins, and then molecules that are produced from the reactions inside the medium and also the cells. So cellular metabolism is closely linked to nutrition and the availability of the nutrients. So cells have several metabolic or biochemical pathways to obtain energy, and then this energy that is formed is now a vital component of the metabolism. So that's how the cell should be viable. So three main purposes of metabolism. So number one, conversion of food to energy to operate the whole cellular process. Next is the conversion of food or fuel to building blocks of protein, lipids, or the micronutrient or the carbohydrates and nucleic acids as well. So the elimination also of nitrogenous waste as well. So cytotoxicity, as discussed earlier, of course, to know if it's toxic to the cell already, or if the compounds are destroying the living cells already. So the cell killing property of a chemical compound is called the cytotoxicity. So of course we know this already. We do this to identify like the harmful substances, and then of course testing these compounds of the viability cells grown in culture is a predictor of potential toxic effects in cells and even animals. So dying cells have highly compromised cellular membranes, which allow the release of cytoplasmic components and penetration of some dyes, okay, so such as fluorescent dyes within the structure or the loss of membrane integrity. So non-radioactive bioassays have been developed to measure cytotoxicity for both proliferating and non-proliferating cell samples. Okay, so we have an example already for the dye. So we have the trypan blue, and then we also can use biomarkers to measure cellular activity or process. Next, it can also be assessed by looking through the cell membrane integrity, or measuring or observing also MMP or mitochondrial activity as well. So we measure it usually using a spectrometer or the absorbance, or nowadays there's a multi reader or microplate reader, so that can measure colorimetric. So it depends on the absorbance of colors, and then fluorescence, and also luminescence as well. So luminometer and fluorometer. So the common methods that is used are this Parmesan dyes such as MTT, which is the very famous one, and then XTT assay. So using also reducing dyes such as Alamar Blue and Presto Blue. So another one, we can also use protease biomarkers and different assays that measures ATP content as well. Okay, we know this already. So just to check the health parameters of the cell, we have the cell viability. We know already how to get that. And then here, so the commercially available MTT and Alamar Blue. So here you can collect or reconstitute the amount that you just need for your experiment. So you should also take note that that there's some effect on its if the dye or the stain. So you need to prepare it in a reconstituted manner. Okay, so are you still there? So okay, MTT assay, it's actually very cheap and mostly widely used here in the Philippines. It has a colorimetric method in quantifying the viable cells, and it's based on this ability of the NADPH dependent cellular oxidoreductase enzyme to produce this MTS tetrazolium that is in yellow color. So after that, if it has an effect with the compound, it will produce a formazine product that is color purple. So these enzymes are present only in active or viable cells. So this MTT assay actually somehow similar with the protocol I already somehow discussed earlier in the lecture part. So it's basically just adding 50 ml of the solution, and then you incubate it for 3 hours, and then after that you add the MTT solvent in each well, and then wrap it in the foil, shake it in a orbital shaker for 15 minutes, and then you can read it with a spectrophotometer or multi-reader at 590 nanometer. So just like this, okay, almost the same. So same thing as other reagents. So this one is from ABB Bioscience. So this one they use rezarurine. So it's a blue and low fluorescence light, and then if there is a viable cell, it will undergo this reaction, and then it will turn pink into high fluorescence, and you just need to read it in a fluorescence reader or multi-reader. So this one, this is MTT assay. So originally it's color yellow. So if it has, if it has cells, so it will somehow turn into, is it kind of purple? Okay. Okay, so usually it should be done in the dark, and because it is sensitive to the light. And then the solubility, of course, of the solution. So make sure that it is shaked well or mixed well. So the solubilization solution usually DMSO or ethanol or SDS in hydro or in HCl hydrochloride is added to dissolve the insoluble purple formazine product into the colored solution. Okay. So the absorbance of this colored solution can be quantified by measuring in spectrum photometer, and then the degree of light absorption depends on the solvent, or for example, DMSO. So for the key points in MTT assay, so the reduction of tetrazolium salts to colored formazine compounds only occurs in metabolically active cells. So the reduction of this is performed through the enzymes, which is the dehydrogenases, and then the lactate dehydrogenase present in the mitochondria of the cell. So actively proliferating cells increase their metabolic activity, while the cells exposed to toxins will have the decreased activity. The mechanism of the other type of cytotoxic dye naman is like for the resazurin. So it's a blue dye, okay, and itself it's weakly fluorescent until it irreversibly reduced to pink colored and highly red fluorescent resorufin. So it is used as an oxidation reduction indicator in the cell viability assays for both aerobic and anaerobic respiration. So example of these are yung Alamar blue that you see earlier, and then the Presto blue. Okay. So next is the Alamar blue. So the active component of Alamar blue is resurin. So as discussed earlier, so in the viable cells, of course, it turns from blue and it reduces to resorufin, which is pink. So which is highly fluorescent, and then the intensity of fluorescence produced is proportional to the number of living cells as well. Okay. So it's just a schematic diagram of how you do it. So you plate it in a 96 well plate, and then you treat it with the with your MTT. Okay, maybe like after 24 hours, you can treat it with your compound, and maybe another 24 hours, you can treat it with your MTT or even Alamar blue, and complete the media with wells. And then just for some time or like incubation, and then you can read it after the incubation time. Okay, so again, so for Alamar blue, it's a non-fluorescent blue dye and then reduced into a fluorescent pink dye or red dye. Okay, so this one is just how the Alamar blue looks like in the colorimetric assay for cell viability. Okay, so take note that these are 1-point or endpoint assays. So the fluorescence readout usually, or even the colorimetric. So after you read the your result, so that would be it. And then most of the time, of course, you have several replicates, usually in triplicates, and then you have also your negative control and positive control and things like that. Okay, so somehow the sensitivity of Alamar blue, I think with the use of fluorescence is a little bit better than maybe like other like MTT assay or other cytotoxic assay. So these are just representation, but there's a lot of cytotoxic or cytotoxicity assay. So for me personally, I prefer something that is not toxic to the cell. So something that you can use or monitor, even though you treat them with the chemical. So you can still monitor it maybe after several days, especially when you're doing time-dependent assays. So, but it all depends on your budget and also depends on the type of experiment or the problem that you wanted to solve, okay, to answer. Next is the C. elegans as a model organism. So here, as you can see, C. elegans studies, or this are the research area or the growth of research area. So the research paper that has been published per year has been increasing since 1965. So up until now, and even the cell elegans laboratories has been increasing, and research area as well. But unfortunately, here in the Philippines, it's not that well studied. I think I just know one expert that is I know of researching um, um, like different topics using C. elegans as their model organism. Next. Okay, are you still there? Anyway, so now you can see here, so C. elegans is a small, free-living, non-parasitic soil nematode. So it survives by feeding on bacteria, so different bacteria. So here you can see how it develops, okay, from the egg and then eventually to the worm. So it looks like this. It has different parts. So you have here his head, or we don't know, or he, or because sometimes it's him hermaphrodite as well. So this is the anterior part, posterior part. So you can see how this one's the slenderer part compared to here. So they have different organs as well that are present. So here, as I said, we have two sexes. So we have an hermaphrodite with a distinct features compared to the male counterpart, which has lesser features and unique organs as well. Okay, so this is just a representation of its life cycle. So from adult that can be a little bit too long, so that are capable also of laying egg, and then it undergoes in utero development. So as you can see here, like from egg to gastrula and to different uh, developmental stages. So it's a good model for developmental biology researches as well. So here, I'm not sure if you can see. So they just um, develop into a dower for up to like hours here, and then after for 4 months, it will develop into like an L4, or somehow like a preteen or before young adult stage, or L4, or it can go actually this way also. So from L1 stage after 12 hours, L2 after 3 hours, and then L3, and then after 8 hours, L4, and then um, it can develop into a young adult and then adult again, laying eggs. So, short history of C. elegans. So, C. elegans is a very promising model. So, it has been developed in 1963 by Brenner, and he also published a lot of mutant, or developed a lot of mutant C. elegans organisms. So, it it was first determined yung mga post-embryonic cell lineages niya by Horwitz in 1976. And then also Horwitz developed the concept of programmed cell death in C. elegans. Okay, so let's see. So the complete connectivity of the nervous system was established in 1986. And then this one, very important discovery was the RNAi and mRNAi discovered in worms, which gained Nobel Prize by Mello. So it was first use of GFP, the green fluorescent parang dye in animals, also gained a Nobel Prize. So there's a lot of Nobel Prize winner with this field. So it's the first animal whose whole genome was sequenced as well. So very promising. So here, as we mentioned, si Horvitz, si Brenner, and Sulston. So the recent Nobel Prize winner in physiology or medicine, and also Fire and Mello on physiology of medicine as well, and Clafley on in 2008. So Brenner was actually the one who shed light on the C. elegans in 1974 pa, with a study on developing the organism, yung mga mutants niya, and all that. And then the worm had many advantages since it has small size, its genome was easily sequenced. That's why it's the first organism to be sequenced. It has a rapid cell cycle, as we have seen sa cell cycle kanina. And then the sexual system of self-fertilization, making it easy to isolate the recessive genes or recessive mutants on the chromosomes that are made favorable organism. So it's good for genetic studies, for behavior studies as well, developmental studies, aging studies, so all that. Okay, let's see. Are you still there? Okay, so just advantages of the C. elegans as a research tool. As I mentioned earlier, it's very simple. It's easy to grow. It has a short life cycle, powerful genetic studies because the sequences are already available, and they both have the self-fertile and the cross-fertile um, sides or areas or fields, and then it's transparent and very small. It has invariant cell lineage, and then fully describe anatomy and development, and so on and so forth. So, yeah, I'm wondering why there's a little or less study of this model here, since it is easy to develop or easy to manage technically. Developmental biology. So these are the applications. So I said earlier. So it's very important in developmental researches and also cell biology. So since it is transparent, it helps to view in the microscope easily, and you can see the development very easily. And then you can also try to stain it with GFP, and the individual cell lineages can be easily traced and directly inspected by light microscope, and programmed cell death or apoptosis can also be visualized using a simple DIC light microscope. So yeah, so here, as I mentioned earlier, so there's like specific cells or cellular architecture can be examined also using reporter genes like GFP, so green fluorescent protein. These ILIS genes or lysozyme exhibit distinct patterns of the pharyngeal expression. So this GFP are small protein derived from the luminous jellyfish and can be introduced by transgenic tag to mark any protein in the animal. Another application is neurobiology. So that's why it's also important for behavioral studies as well. So it acts as a model for neuronal development and function. So no brain per se, but have sophisticated nervous system that has 302 neurons and 259 cells. So C. elegans respond to chemoattractants and repellents. So one example, for example, is like there's a chemical, and then you can see how they respond to the chemoattractants or repellents. So you can observe them in the Petri dishes or Petri plates. Then also other studies are like laser beams that selectively, like they can selectively cut the neurons, and electrophysiology studies can be conducted using that type of experiments. And then connectome, the complete wiring diagram can also be, that also has been established as well for neurobio experiments. Next is aging. So since the life cycle I mentioned is very short, so it helps to conduct genetic screens to find longevity genes as well. And then it has been used also for human disease studies or as a model for human disease. So 75% of the human disease genes have potential C. elegans homologs. So 40 to 50% have C. elegans ortholog as well. Okay, so the last part, do we still have time? Meron pa naman, no? Okay. So zebrafish as a model organism. So zebrafish is a tropical native fish or in the Southeast Asia. Usually here you can see the male. Okay, the male here on top is more slender compared to the female. And then the life cycle is around 12 weeks. And then the embryo is around 0.7 millimeter and it's transparent. So the egg is similar of that of the chicken, and embryos are developed externally, which allows easy observation. So here, they also have a complete genome sequences as well. And the advantages of using this research tool is it's relatively cheaper. So it's also an in vivo model where it's cheaper compared to other in vivo models such as mice or rats. And it occupies much less space. So maybe you just need also to, you can also develop it into a DIY garage lab. So you can make like your aquariums, and then that usually occupies not that a lot of space. So it has high fecundity and transparent embryos laying 200 to 300 eggs every 1 week or 5 to 7 days if maintained properly. So the eggs remain transparent from fertilization to when the tissues has become dense and pigmentation is initiated. So this one, you can also study more about, for example, their genetics as well, like pigmentation or its characteristics as well. So experiments has been standardized using zebrafish or Daniel as a model. So the embryo, so most of it also is also developmental. Toxicity test is also available or it can be done, and alternative to fish acute toxicity test. So which is because a fish acute toxicity test is a bit more in terms of space, it requires more space. So next is primary and immortalized cell lines. So from this zebrafish or Laniarero has been developed from adult and embryonic zebrafish. Okay, so you can have a in vitro counterpart of this as well. So if you want to screen it sa in vitro muna, and then you can use zebrafish as your in vivo. Next is the mutagenicity, or these are the applications or experiments that you can do. Mutagenicity test or teratogenicity test, developmental studies, endocrine disruption. So like, for example, if you know like different EDCs, endocrine disrupting chemicals such as BPA, PFOA, what else? BPS, phthalates, all those endocrine disrupting chemicals that mimics your hormones or your endocrine molecules. And then next also is identifying toxicological hotspots in ecological studies. You can also use this as a combination of genetics and vertebrate biology, and has a great potential for cancer model as well. And a lot of researchers also on aging and osteoporosis as well. So you can see here a sample diagram from Padilla et al. So here, for how many hours? Maybe 144 hours. So from fertilization to like single embryo, like from the embryo stage, you get like the eggs here, and then you treat it with a chemical, and then you somehow change, just like cells, you change the media, change the water, and then treatment as well. So while you change, and then last is this one, replacing it to a buffer, and then you check if the larva is alive or not, and then you just record it. So something like that, and then you assess you can assess their features. Okay, so just like this. So sample study here is the toxic effect of FENA. This is an EDC, or perfluorononoic acid. So there's a lot of PFCs that are available in their environment that we have exposure every day. So here you can see this one are the control group. So this is how it usually look like from 24 hours, 48 hours, 72, and 96. And then here with different concentration of PFNA. So here the highest concentration, as you can see, how the zebrafish look like. So somehow it has some developmental defects that have occurred. Okay, so even like delayed, maybe delayed development of some sort, or some defects in the earlier or 48 hours part. Okay, so these are the regular or the usual developmental defects. So sometimes you can see irregular fins, missing fins, stunt fins, arcardia, bradycardia, so like the heartbeat. So you can easily see that in a microscope, dissecting microscope, and check the beat. Or sometimes you can use video for behavioral assays. Here you can also check cardiac enlargement, tube heart, brachycephalic. So as you can see earlier, like a lot of like enlargement of different part of the zebrafish, excuse me, microcephalic, and then enlarged organs, granular organs, hemorrhage, lumps or tumors, jaundice. There's a kink in the tail, lordosis, scoliosis, eel-like body, and emancipated. So many more. And then there's a lot of studies also regarding like yeah, behavioral, or you can also do like also like mental studies like anxiety, or like different yeah neuronal studies as well. Okay, so I think, oh, we finished early. Actually, I have more, but this one, I think I'll just give it to the genetics part, so mutageneticity assay. So there's a lot of simple experiments such as what's that test? Allium cepa test or assay, or other mutagenetic or AMIS test. So this one maybe yeah, you can discuss it. I can just send it to you, and you will somehow know already the steps, or just a brief schematic methodology that can be done in your institute. Okay. So are there any other questions? So after this, we will have an activity of the, what's that? On assessing some research projects. So maybe now you have an idea of some researches that maybe you can do at home, right? Maybe you can do in your garage or in your DIY garage. But of course, be careful. Of course, you should always be have a biosafety in mind. Okay. So if it's doable, maybe it's you can consult some experts in your town or somewhere near your area, in your colleagues or something like that. Okay, so if there are any questions, maybe I can take in early.
Sige po, ma'am. Thank you po, ma'am, for sharing with us your knowledge on bioassay techniques. So I think we can proceed already with our open forum to answer some of your questions. So again, same with earlier's open forum, you simply need to type your questions either in the comment section of the YouTube live streaming or in the chat feature or chat box for our Zoom participants. So ma'am, we already have 3 questions po from Minette Eloreta. So unahin ko na lang po muna yung first. So first question niya po ay, can we use natural seawater? So I think ma'am, this is in relation po doon sa brine shrimp
Okay.
lethality assay.
Okay, so that's why we use artificial water, because we want to have a controlled environment. So the natural water, salt water, it can be used, but we don't know its properties, and sometimes we don't know if it has like microorganism or other debris or parts that are not good for the growth of the brine shrimp. Okay, so it might compromise your experiment as well. So it's better. It's just easy to have your distilled water and then rock salt. So just for a uniform controlled environment.
Thank you po, ma'am. So for her second question po, can we be given an idea of the cost per assay?
Of what assay?
Per assay po, ma'am. So I think in
So all?
general, yes po, ma'am.
Okay. So I'm not really sure. It depends on the facility and if they are offering that kind of assay. So for what I know so far is MTT assay. So one plate is 8,000. So you can have 96 well, actually just or 60 wells. So you can somehow check different concentrations or different what's that, extracts as well. So that one is the fee of MCCL in UP Diliman way back maybe 2019. So I'm not sure the recent rates are. So but it's like that, 8,000 per plate.
Thank you po,
For
ma'am.
the other test, brine shrimp, I'm not really sure kung how much, but maybe you can do it on your own, right? It's easily available, like in the pet shop. So maybe you can do it in your own garage or own home or own institute. If you have some space in your institute, can easily adapt it, especially for the high school institutes or STEM students or something like that. And then what other? I think for C elegans, I only know one facility in UP Manila, but I'm not sure about their rates. What you can also do is maybe develop your own. You can ask them some of the samples, but I'm not sure if they will allow. And then Daniel Rerio, CLSU has some. You can get clones from there, or like you can get like a fresh embryos or eggs. And I'm not sure if they have service facility for that, but it's where we usually get Daniel Rerio.
So thank you again po, ma'am. Thank you po. So bali doon naman na po tayo sa last question po ni Miss Minette Eloreta. Second to the last question po ni Ma'am Minette. So for the third question of Ma'am Minette, I think this is in relation po, ma'am, doon sa scientific name ng C. elegans. So is C. elegans referring to Caenorhabditis elegans?
Yes.
Thank you po, ma'am. Now for the next or rather last question po of Ma'am Minette. Aside from human errors, what are some problems encountered in doing assays and how to resolve it?
Okay, so you mean like all the assays? I think, of course, what's that? The equipment or the reagents that you have also. So you have to consider a lot of things, especially as we discussed earlier. The physical or the environment should be similar or somehow it mimics their natural environment. So, and also it should be controlled so that you won't consider other factors in terms of comparing it with like for example control or other environmental factors as well. What else? So anyways, it takes also expertise and time to develop a certain skill also in optimizing even the setup. So sometimes your experiment will not be reproducible, but if you have to really consider all the factors affecting your experiment, the environment, the handling, also the location, the place. So you need to consider all that. Okay. Yes.
Ma'am, we have another question po from Chris Paul Pagawa, from Chris Paul Pagawa. Can we use BSLA for the preliminary investigation of plant extracts for their anti-cancer potential? Do you also offer any laboratory services in UPB, in UP Baguio with these assays discussed?
Okay, I'm glad you asked that, right? Okay, so for this first question, for anti-cancer potential, I think it will be good for BSL. This is lethality assay, so it will be good for cytotoxicity screening. So somehow you can relate it, like for the proliferative or its screening or effects on sa like in vivo model, so for the initial screening. But for anti-cancer, I'm not yet sure of the specific. It should not be maybe lethality assay. Maybe there's another um methods for that or other assays for that. And then for the laboratory services, so we have laboratory services in UPB. We have a newly established SRC, Science Research Center. But then again, these assay, I don't think it's available for now, but we're still on the process or on the works of establishing the research center. So hopefully in the future, we'll be able to um, to offer you the services that you are that you needed. So currently we do have service facilities. So maybe you can get back to me on the available facilities or service that we offer in the Science Research Center at this point. So yeah, that's it.
Thank you, ma'am. So we have another question po from Jim Carlo Pula. So for the brine shrimp assay, what can you suggest as an alternative way to isolate or count or transfer the brine shrimps to another container like test tube?
Oh, okay. So like another container. So I think like a glass. So as long as it's clean or somehow, of course we don't have autoclave in our house, right? So maybe you can use some kind of what's that? Like a what do you call this? The glass container, like a garapon or something like that, or like different small containers, and then that can be pressurized. Like you can aseptically clean it using a pressure cooker. So just like your autoclave. So yeah, you can consider that as an alternative container. Yeah, mason jar. Yeah, that's right. Correct.
Thank you po, ma'am. So, ayan. So, while waiting for other questions, ma'am, maisingit ko lang po.
Mm-hmm.
Ma'am, regarding the brine shrimp lethality assay, I would like to ask din po sana, I'm just curious din po, ma'am. Regarding po doon sa room temperature part po ng brine shrimp lethality assay, would there be a difference po if ever na ginawa po siya on a lowland area kung saan mainit po to a setup being performed in UP Baguio na malamig-lamig po, since yun nga po ay yung room temperature po nila would differ? If ever po, ma'am, just curious po, kung magkakaroon po ba yun ng big difference if ever?
Yeah, it would make sense. So room temperature is usually 24 degrees or 20 to 24 degrees. So it of course temperature is a big what's that requirement for not only microorganisms, not just cells, but also for organisms such as brine shrimp. So there would be differences, but I don't think it depends on how, like, the plus and minus degree Celsius that is different. But I think plus and minus two, but I'm not really sure on this. So I'll maybe have to get back to you on that question. But yeah, it would definitely affect or there would be a difference in maybe the results. But if you have your like for example, control. So if you just test it across your control or test it and compare it across your control, it might be okay. So depending on the circumstances.
Thank you so much, ma'am, for the clarification. Thank you, ma'am. So I think that's it. Wala na po tayong pumapasok na questions. So thank you once again, ma'am, and thank you once again to our participants for raising your questions and other concerns. Now, as per schedule, of course, after this, we are going to have a 15-minute break first before we proceed to our short activity. But before we take our merienda or our CR break po, just a few reminders. So first, we would like to remind once again our participants to please answer the evaluation form later, which will be forwarded as a link in the comment section in the YouTube live streaming, or will be posted directly to the chat feature of this Zoom meeting. Also, in particular to our Zoom participants, immediately after our 15-minute break po, you will be directed to a particular breakout room. Okay? And then once you enter that particular breakout room, we or you will proceed to your short activity on assessing research proposals. And then immediately after the short activity po, wag na lang po muna tayo agad mag-leave sa Zoom meeting as we may need to return to this main room for some announcements po. Now for our viewers viewing this live via YouTube live streaming, unfortunately po, after this open forum, wala na po tayong live streaming, kung kaya hindi na po natin masasaksihan yung mga mangyayari po sa ating breakout rooms. But still, for you to benefit of the activity, you have two options po. So first, you may fully use the FB platform to access certain files and to raise certain questions, which will be answered immediately right there and then. And for our second option po, for our YouTube viewers or YouTube participants, you may simply attend this Zoom meeting as this Zoom meeting may still accommodate more participants. So that would be all. I think we can all have our break, and may we please ask our participants to be back after 15 minutes. And so we can all have our break. Okay. Thank you once again.
Okay, hello po. Good afternoon, everyone. So for those people who are able to switch on their videos, kindly switch on your videos po muna para naman po may interaction tayo this afternoon. Ayan, sana po nakapagmerienda po tayo. So, kumusta po muna tayo? For those people na nandito pa sa amin via Zoom and kung meron pa nanonood via the YouTube live stream, press X naman dyan sa ating mga chat button. I-send nyo po. Ayan. Ayan. Sana po hindi rin po kayo masyadong nasistress sa sobrang init ng panahon. Nandito po kami ngayon sa Baguio pero mainit na po. So, sana hindi po kayo masyadong nasistress. And I hope may nakuha po kayong information para sa inyong mga gagamiting resources para sa inyong pong pagtuturo this coming classes. So thank you very much po. So a few reminders po before we proceed with the next part of the activity. You will be divided into breakout rooms po and we have assigned you randomly already. Gagawin po yan ng ating SINSM Tech and Secretariat team. So you will be assigned to a specific SINSM facilitator that will help you with the breakout session activity. So mamaya po, once we start the breakout rooms, wag po kayong magugulat if bigla pong magba-black yung screen and you will be redirected to another breakout room. So do not click anything po. Also in your breakout rooms, there will be tech people that will be assisting for your tech concerns if you have tech concerns. And you will see the main facilitator for your breakout rooms. Nakalagay po yung SINSM facilitator and their names. So just to introduce our 5 facilitators, we have Professor Rosel Amor Indong, we have Professor Romeo Dizon, we have Professor Meiji Baganga, we have Professor Ronnie Kalugay, and we have Miss Kimberly Paglingayan. So sila po yung ating 5 activity facilitators for this afternoon. So I hope everyone's excited. Sana po ready po kayo to share your insights. This is your avenue to ask questions to our amazing panel of facilitators. If you have concerns about conducting research, they're the best people to ask and also consult with regarding your concerns, especially right now where it's difficult to do your own research proposals in your own research projects. So without further ado, SINSM Tech team, kindly bring us to our breakout rooms. So hello everyone, welcome back to our SINSM main room. Ayan, habang hinihintay natin na makabalik pa yung iba nating participants in our main room. Some reminders for our participants before we end this day. What is being shown in your screen right now is our evaluation form for day 1. So this is our survey form. So you can either scan the QR code or you can type in the link which is shown in your screen for the evaluation form. This is very important because this will be our basis for giving your certificates after this event. So please kindly answer the survey form for day 1, including our speakers for this day. Also, we would like to thank our SINSM facilitators. Maraming salamat po. For sure, marami po kayong nabigyan ng inspiration at marami po kayo nabigay na insights pagdating sa kanilang mga research projects. We would like to thank Professor Rosel Amor Indong. Ayan, napakaganda ni ma'am, diba? At saka napakatalino. We have to thank, of course, Professor Ronnie Kalugay. Ayan, napakadaming research experience. Professor Rom Dizon, Professor Meiji Bagangaw, and Professor Kimberly Paglingayan. Thank you very much. We'll try our best to, give lines of communication for them in case you have other questions or in case you need consultation, particularly for some aspects of your research project. So we will be very glad to help you with that. Again, to all of our SINSM facilitators for the breakout sessions, thank you very much po. Other reminders, again, we have to remind all of our participants that you need to attend at least 2 out of the 3 plenary sessions and activities for SINSM for you to have a certificate. So again, please make sure that you can attend so that you will receive later on a certificate from the SINSM Secretariat. Other reminders po natin, again, tomorrow is another day for the biology discipline. So posted na po sa ating Facebook page, sa SINSM Facebook page and sa ating official Facebook group yung line-up for the activities tomorrow. So we have another speaker for tomorrow. Nandun na po sa ating Facebook page. So if you're not yet a member of the official Facebook group for biology participants, kindly visit the link in your chat box on Zoom right now so you can join the official Facebook group. So we'll be posting updates and announcements on that Facebook group. So thank you very much po sa join in advance. And lastly, again, please, we'll repeat this, answer the evaluation form. This will be very helpful in improving our activities and our sessions, future sessions for SINSM. So thank you very much. So may mga energy pa po ba tayo ngayon? Kung may energy pa po tayo ngayon, pakilagay naman po sa chat box yung ulam nyo kaninang lunch. Kung kaya nyo pa pong i-chat, pakilagay naman po kung ano yung ulam nyo kanina nung lunch. Gusto lang po namin malaman dahil hindi pa po kami kumakain. No, kidding. Again, thank you very much for this wonderful first day. It's been an honor to be, moderating and, helping our participants in this amazing activity. Nakakagutom yung nilagang pork at saka sinigang and sisig. Talagang papunta na po tayong karinderya, ganyan. So maraming salamat. Again, kindly finish our evaluation form for today. We will be looking forward to all of your insights so that we can improve our activity and we will see you on Facebook. So pwede po tayong mag-communicate there. Pwede po namin maybe ibigay yung mga official emails ng ating mga facilitators so that you can contact them. So, and also the resources na napakita yung mga PowerPoint presentations, we will provide them once ready na po yung mga links for that. So again, thank you very much for everyone. Maraming salamat sa ating participants. Maraming salamat po sa SINSM Tech Team. And maraming salamat po sa Secretariat and sa ating mga facilitators. See you all tomorrow morning. Maaga po tayo sa Zoom. Meron din po tayong live stream link again tomorrow. So, kita-kita po tomorrow. Maraming salamat. Enjoy your afternoon. Maraming salamat po.
Bukas ha, mag-unmute tayo. Yay! Ay,
Hello to our participants. You may now leave the Zoom meeting po if nakuha niyo na yung links. So in case there are other questions for the event tomorrow, you can contact us via the Facebook group. Sasagot po kami sa mga concerns niyo doon. You can comment on the thread that we're going to form mamaya po gabi. So maraming salamat po. Thank you very much din po sa pag-attend at pag-participate. Thank you very much po.